or the cell membrane. Look at this structure. It is a cell. What is the cell membrane? That means the outermost boundary or the outermost structure of the cell is called as the cell membrane or the plasma membrane. So first of all, in the 1950, the electron microscope was discovered. So after the electron microscope discovery, then only the discovery of the plasma membrane was done. And the first discovery of the plasma membrane was done in the cell that is called as the RBC, or the red blood cell. That means in the RBC, the first analysis of the cell membrane was done. So now look at this structure. This is the cell membrane structure. So if I zoom this person, it looks like this. That means actually the plasma membrane is not a single layer structure, it is a two layer structure. This is the one layer, this is the second layer. What is called as the leaflet. Leaflet. That means two leaflet. This is the one leaflet, this is the second leaflet. Or it is called as the one layer or second layer. So always, that's why the cell membrane is double membrane structure. So now come to the structure of the plasma membrane. What is the constituents of the plasma membrane? So in the plasma membrane, mainly the lipid and protein are present. Okay. So this structure is called as the lipid. This is the one structure, this is the one lipid, this is another lipid, this is third lipid. So now come to the what is the structure of the actual lipid. The one lipid structure is looked like this. That means it has two segments. One is called as the head, it is called as the head part, and it is called as the tail part. So head part always the hydrophilic. What is the hydrophilic? The structure which always wants to interact with the water molecule is called as the hydrophilic. So what is the tail? Tail is the hydrophobic structure. Hydrophobic structure. That means it is the same structure in which both hydrophobic and the hydrophilic structure are present. So that's why the lipid is called as the amphipathic molecule. Amphipathic molecule. So what is the amphipathic molecule? That means any structure which contains both hydrophilic as well as hydrophobic structure, that is called as the amphipathic molecule. So now in the lipid, in the head part, it contains glycerin along with phosphate. So this is the head part. And in the tail part, this is the one tail, this is the another tail. So what is this tail part? These are called as the fatty acid. So now, what is the fatty acid? Fatty acid is simply the chain of the hydrocarbons, and at the last part, COOH group is attached. So this is called as the fatty acid. The fatty acid can be two types. One is called as the saturated fatty acid, and another is the unsaturated fatty acid. So what is the saturated fatty acid? That means in the fatty acid, if all the bonds are the single bond, then it is called as the saturated fatty acid. So this type of the structure of the fatty acid is called as the saturated fatty acid. So now look at what is the unsaturated fatty acid. That means in the fatty acid, if at least one double bond is present, or more than one double bond is present, then it is called as the unsaturated fatty acid. Okay. So now you know what is the saturated fatty acid, what is the unsaturated fatty acid. So look at the structure of the, so what is the lipid? The lipid is the, at the head part, the glycerol and the phosphate. Look at the structure. This is the glycerol structure, this is the phosphate structure. And this is the tail. So tail means it is the only the fatty acid. So lipid means glycerol, phosphate plus fatty acid. So one is the saturated fatty acid, another is the unsaturated fatty acid. The saturated fatty acid, it actually present like this. That means in a straight line structure. But in the unsaturated fatty acid, because of the presence of the double bond structure, it bends somewhere it bends, or it is called as a kink. K-I-N-K, kink. So what is the kink? That means simply the bending. That means if the saturated fatty acid is present in the lipid, then it looks like this. If one unsaturated fatty acid is present, then it looks like this, or it is designated as this. So always, normally in the membrane, the lipid contains one unsaturated fatty acid. So that's why if we look at the book, then it looks like this. That means one tail is bending like this. Here looks like this. One is simple straight, another is bending. Why it is bending? It is called as the kink. That means in that area, a double bond structure is present and the whole fatty acid is unsaturated fatty acid. Okay, so plasma membrane contains protein plus lipid. So actually, which type of the lipid is present? So normally, or in the plasma membrane, the lipid is phospholipid. Phospholipid. What is the phospholipid? That means the lipid in which the head part contains a phosphate group. Here the PO4. A phosphate group is present. That's why it is called as a phospholipid. Okay. So always in the plasma membrane, the lipid is the phospholipid, and another lipid is present. That is called as the sterol. So what is the sterol? Sterol is a subgroup of steroid. Subgroup of steroid, or the group, one of the group of the steroid is called as the sterol. So different types of the sterol are present in different organisms. For example, in the animal, the sterol is present, that is called as the cholesterol. In the plant, the sterol is present in the plant, is called as the phytosterol. Phytosterol. In the fungi, the sterol is called as the mycosterol. So in different organisms, different types of the sterols are present. So always in the plasma membrane structure, the lipid is present. And which type of the lipid? One is the phospholipid, another is the sterol. So two types of the lipid are present in the plasma membrane. So now come to the protein. Along with the lipid, some proteins are present here. In between that, some proteins are present. So this is all about the structure of the plasma membrane. So now come to the models of plasma membrane. So what is the models of the plasma membrane? That means the plasma membrane, or what are the sequence of the discovery of the plasma membrane. The first model was discovered and it was called as lipid and lipid bilayer model. It was discovered in 1925. Okay, and it was discovered by Gotter and Grendel. Gotter and Grendel. So what is the lipid and the lipid bilayer structure? This structure is called as the lipid and lipid bilayer model, which was discovered in 1925 by Gotter and the Grendel. What is the lipid and the lipid bilayer? So Gotter and the Grendel, they speculated at that time that only the lipid are present in the plasma membrane. This is the one layer, that is the lipid layer. This is the another lipid layer. That means there is no protein, there is no carbohydrate. Only the lipid are present according to that model, which is wrong. But at that time, actually it was the first model. So that's why they thought that it may contain only the lipid layer. That's why it is called the lipid and the lipid bilayer. That means it is a bilayer structure, and the always means in all areas lipid are only present. That means no protein, no carbohydrate is present in that plasma membrane. So the second model is called a sandwich model. Sandwich model. It was discovered in 1935 by Danieli and Debson. So what is the sandwich model? It is the sandwich model. This type of the structure is called as a sandwich model, which was discovered in 1935. So according to this model, here at the outer side the protein is present. Here it is the one protein layer, here it is the one protein layer. And then the inside the lipid layer is present. That means it is called as a sandwich model. That means the lipids are sandwiched by the two protein layers. That means inside the lipid are present, and this lipid layer is sandwiched by protein. That means the outer here the protein, here it is the also the outer protein. That means two proteins, this sandwich the lipid structure. So this is the second model. The third model is called as unit membrane model. Unit membrane model, and it was discovered in 1953. And I have already told that the first electron microscope was discovered in 1950. So, and it was discovered by 1953. That means it is the first model which was discovered after the discovery of the electron microscope. So that is called the unit membrane model. And it was discovered by Robertson. Okay. So what is the unit membrane model? According to that, here the protein layer, this is the unit membrane model. So according to that, the protein layer, it contains protein and lipid, according to this model. And the protein is asymmetric. That means not same in the both layer. That means at the outer layer, look at this structure. It is a thick. And at the inner layer, this is the thin structure. The thin layer of the protein is present, and inside it the lipid is present, according to this model. So that is called the unit membrane model. So the fourth one is most important. That is called as the fluid mosaic model. Fluid mosaic model. And it was discovered in 1972 by Singer and Nicholson. Okay. So please note that the fluid mosaic model, which was discovered in 1972, it is the most accepted theory. It is the most accepted theory of the plasma membrane. That means Even nowadays, we also accept the fluid mosaic model. It is the latest model and it was accepted only about the plasma structure. So that's why if it in the exam, if it will be asked that which is the most accepted theory or which is the accepted theory or which is the latest theory about the plasma model, and the answer will be the fluid mosaic model. And it was discovered by Singer and the Nicholson, and this is the structure of the fluid mosaic model. That means nowadays, whatever we know about the plasma, it is according to the fluid mosaic model. So now come to the extensive study about the fluid mosaic model. So this is the fluid mosaic model. According to that, the plasma membrane contains lipid plus protein plus carbohydrate. Okay. So what type of the lipid are present? One is called as the phospholipid. Another is called as the sterol. So now look at the structure. This is the one layer, the one leaflet. This is the another leaflet. So this structure is called as the lipid structure, and this orange color structure, it is called as the phospholipid. Okay. So the yellow color structure or this structure or here this structure is called as the sterol. Here it is called as the cholesterol. Why it is called as the cholesterol? Because it is the animal cell. This is the animal cell. Why? Because in the animal, the sterol is called as the cholesterol. If it is the plant, the sterol is the phytosterol. If it is the fungi, the sterol is called as the mycosterol. So one is the phospholipid and the sterol. So if in the exam it will be asked that what are the types of the lipid present in the plasma membrane, and it is called as the simply phospholipid and the sterol. And what is the function of the sterol? It helps in the fluidity of the plasma membrane. Fluidity of plasma membrane. Okay. So this is the phospholipid. Phospholipid, that means this type of the structure is called as the phospholipid. One is called as the head part, and it is the hydrophilic, and it is the tail part, and it is the hydrophobic. Now, what do you think that why the head part is called as the hydrophilic? Or why the head part is hydrophilic and why the tail part is hydrophobic? Look at the structure. Let's say this is the cell and this is the membrane of the structure. So that means it looks like this. That means here one is the head part, here at the outer side the head part is present, and then the inside it or the middle like this, here the hydrophobic group, the hydrophobic tail group is present. But why? Always in the cell, let's say this is the one cell, this is the another cell, this is the another cell, this is called as the extracellular matrix. Extracellular matrix. And the inside it, or the inside of the cell is called as the cytosol. So always in the extracellular matrix of the cytosol, the watery condition, the watery solution is present. That means the cytosol is simply the watery condition. So that's why the inside, the water molecule, let's say water molecule, not water, the watery solution or the hydrophilic solution is present. At the outside also, this is called as the extracellular matrix. Here also the hydrophilic or the watery molecules are present. So that's why the head part is hydrophilic, so that it can interact with the water molecule. So always the hydrophilic, they want to interact with the water molecule. What is the hydrophobic? They always want to away from the water molecule. So that's why at the middle part, here the hydrophobic or the tail part is present. At the outer side or the inner side, always the head part is present so that it can interact with the water molecule. In the middle or the core part, it doesn't want to interact with the water molecules. So that's why they always want to away from the water molecules. That's why they are present at the inside of the cell. So this is the phospholipid. This is the sterol which helps in the fluidity of the plasma membrane. So another type of the lipid is also present, that is called as the glycolipid. Glycolipid. So now sorry. So one is the lipid part. So in the lipid part, one is the phospholipid, another is the sterol, another is the glycolipid. What is the glycolipid? We will know after some time. Okay, this is the lipid part. So in the plasma membrane, lipid plus protein is also present. So what is the protein? So here the blue color structure is called as the protein. Here it is the one protein, here another protein, here the another protein. So the proteins are two types. One is called as the peripheral. Another is called as the integral. The peripheral is also called as the extrinsic protein, or the integral protein is also called as the intrinsic protein. So what is the extrinsic, what is the intrinsic protein? So extrinsic, now for the peripheral, as the name suggests, that means it is only present at the periphery. Look at the structure of the protein. This is present only at the outer area of the lipid, and this is called as the extrinsic of the peripheral. So what is the integral? That means it must have gone to the core of the plasma membrane. So look at the structure. It already buried in the core of the plasma membrane. That's why it is the integral or the intrinsic protein, and this is called as the peripheral or extrinsic protein. Okay, so because the peripheral protein, they are present only at the outer side, it binds the lipid membrane very loosely bound. And the integral protein, because it is already buried to the middle of the bilayer, it is strongly bound to the lipid bilayer. Okay, so this is the structure of the lipid. Another lipid, okay, one is the peripheral, another is integral. Some protein like this is also a protein. It already present at the inside and the outside. That means it already buried whole part of the lipid bilayer, and this type of the protein is called as the transmembrane protein. So normally three types of the protein are present. One is the peripheral, which is present at the just outside at the periphery of the lipid bilayer. One is the integral, that means it is already present near the core of the plasma membrane. Another is the transmembrane, which buried along the plasma membrane. That is called the transmembrane protein. So another structure is called as the carbohydrate. Carbohydrate. So the carbohydrate looks like this type of the protein, this type of the carbohydrate. Carbohydrate are simply two types. One is called as the glycoprotein. Another is the glycolipid, which is already I have told. So what is the glycoprotein? What is the glycolipid? So always carbohydrate always present at the outer side. That means either this side or this side. It is not present at the middle of the plasma membrane. And carbohydrate is only present by binding either the protein or the lipid. So glyco means carbohydrate. And the protein means protein. Here it is the glyco means carbohydrate or carb, the lipid. So that means some carbohydrate binds to protein and it is called as a glycoprotein. And some carbohydrate are present by binding with the lipid, and this is called as the glycolipid structure. Look at this structure. This is called as the glycoprotein. That means this is a protein. To this protein a carbohydrate group is attached, and this carbohydrate is called as the glycoprotein. And some protein are present or bound to the lipid structure, and that is called the glycolipid structure. So in summary, in the plasma membrane, lipid plus protein plus carbohydrate are present. Then the lipid, one is the phospholipid and the sterol, they are present in the plasma membrane. Another is the glycolipid is present, which is present at the outer side by binding with the carbohydrate. Another is the protein. There are three types of the protein. One is the peripheral protein, which is present at the periphery. Another is the integral, which is buried in the integral part of the plasma membrane. Another is the transmembrane. That means it already buried to the whole plasma membrane structure. That is called the transmembrane protein structure. Another is called the carbohydrate. Carbohydrate is present only at the outer side by binding with the lipid and the protein. Lipid is bind to the protein, then it is called as the glycoprotein. If it is bound to the lipid, then it is called as the glycolipid. So this is all about the plasma membrane structure. So now in some different cell, different types of the composition of the lipid and the protein and the carbohydrate present. And the exam always asks what are the constituents or the composition of the lipid and the protein and the carbohydrate present in the RBC. Why it is asked in the RBC? Because the first discovery of the plasma membrane was done in the RBC. So in the RBC, the 52% lipid are present, 52% protein are present, 40% lipid are present, and 10% carbohydrate are present. Okay, so this is the structure of the lipid bilayer. So one is called as the, why it is called the fluid mosaic model. The structure is called as the fluid mosaic model. One is fluid, another is the mosaic. Mosaic means like the tiles. The mosaic The fluid is due to the lipid structure, and the mosaic is due to the protein plus sterol. What is the mosaic? That means like the tile structure. If in the house, then the tile house, the tiles are present at the house like this. So this is called the mosaic. Here also, the lipid and the protein and the sterol are present like the mosaic model. That's why it is called the mosaic. And why it is called the fluid? And the fluid is due to the lipid. Why it is called the fluid? That means the plasma membrane is not very strong or not very hard or not very rigid. It is very, very soft. Okay. So unlike the cell wall is very hard or the very rigid, which is present at the plant cell. That means in the plant cell, this is the plant cell, this is the plasma membrane, and the outer side of the plasma membrane, another layer is present, and it is called as the cell wall. The cell wall is very hard, but the plasma membrane is not very hard. It is the fluid-like structure or the very soft. Why it is called as a fluid-like structure? Because it always rotates around each other. That means it is not fixed. That means let's say this is the lipid bilayer structure. It is always not fixed. It is always in a movement like structure. That means always it is moving. The structure is always moving. And that's why it is called the fluid structure. So why or how the fluid or how the lipid is moved? Lipid moves by two methods. One is called as the flip flop. Another is called as the lateral movement. Lateral movement. If movement of the lipid, there are two types of the movement of the lipid. One is called as the flip flop. Another is the lateral movement. What is the flip flop? Look at the structure. If this lipid, it comes to this layer, and this lipid comes to this layer, this is called as the flip-flop movement. That means from the one leaflet to another leaflet, or from one layer to another layer, then it is called as a flip-flop. Or simply, it is a vertical movement. That means from one leaflet to another leaflet. What is the lateral movement? That means if this lipid, it comes to here, and here it comes to here, then only it is called as a lateral movement or the horizontal movement. Horizontal movement. So always in the plasma membrane, it is not fixed. They move always around each other. So that's why the lateral movement of the protein, lateral movement of the lipid, it is responsible for the fluidity of the plasma membrane, along with the cholesterol. Previously we have already studied about the sterol. That means the sterol, it maintains the fluidity of the plasma membrane. For the lateral movement of the lipid, it is responsible for the fluidity of the plasma membrane. So along like the lipid, the protein, let's say in the inside, some protein are present. The protein are also moved, but only in the lateral movement. That means the protein can move only laterally. That means in one leaflet only. But it cannot move like the flip-flop. That means the protein cannot go from the one layer to another layer. But the lipid can. So lipid can move from the flip-flop or the lateral movement. That means lipid can move horizontally as well as vertically. The protein can be moved by the laterally. That means in same leaflet or the same layer, it can go or it can move, but in the horizontally, in the vertically, they cannot move. So this is the movement of the protein in the lipid. So now come to the function of the plasma membrane. What is the function of the plasma membrane? So what is the function of plasma membrane? One of the function is called as the cell growth. Cell growth. Cell growth means, let's say this is a one cell, it grows like this. Why? Because the plasma membrane is fluid, not hard. Let's say this is, if it is the hard like cell wall, then the growth of the cell cannot be done. So that's why one of the function of the plasma membrane is the cell growth, another is cell division. Cell division. Cell division means one cell divides to two cells, or the mitosis and the meiosis. You will know about after some time. So the mitosis, the meiosis, two types of the cell division is present in the cell, and the cell division is also due to the plasma membrane. Another is endocytosis. Endocytosis. What is the endocytosis? Let's say this is the cell. Some molecule is present outside the cell. It enters into the cell and it is called the endocytosis, or the engulfment of the cell, or engulfment of the molecule by the cell is called as the endocytosis, and it is also done by the plasma membrane because the plasma membrane is fluid or the soft. And the fourth one is intercellular junction, formation of intercellular junction. What is the intercellular junction? Let's say this is the one cell, this is the another cell, this is the another cell. So here the cell junction, the cell interaction is also helped by the plasma membrane. This is the function of the plasma membrane. Now come to the transport of the plasma membrane. Transport across plasma membrane. So what is the plasma membrane which helps in the transport across? So always the plasma membrane, it is a gatekeeper. It acts as a gatekeeper. That means, let's say it is a cell. It is a gatekeeper. That means it permits the molecule by seeing what is the composition of this molecule. This is called the gatekeeper. That means it selectively allows some molecule. So that's why the plasma membrane is also called as the selectively permeable membrane. Permeable membrane. Or it is also called as the plasma membrane is also called as the semi-permeable membrane. Semi-permeable membrane. That means this membrane or the plasma membrane structure, it doesn't allow to all the molecules. It selectively allows some molecule. That's why it is called as the selectively permeable membrane or the semi-permeable membrane. So what are the different types of the transport across the plasma membrane? There are two types of the transport. The present. One is called as the passive transport. Passive transport. Another is the active transport. So what is the passive transport? The passive transport means that any molecule which can transport without use of the energy. Without use of energy. Or the active transport, it transport to the cell or the outside the cell by using the energy. Using the energy. Now come to the passive transport. The passive transport means the molecule transports along the concentration gradient. Along concentration gradient. What is the along concentration gradient? That means from the higher concentration to lower concentration. Look at this structure. Let's say this is the outer surface, outer layer. This is the inner layer. What is the outer layer? That means let's say this is the cell. It is the inner, it is the outer. Let's say this structure, if it is zoomed, it looks like this. That means it is the outer layer, it is the inner layer. Look at this molecule. This molecule is present higher concentration at the outer side and the lower concentration in the lower side. That means if the two molecules are present in the inside, in the 1, 2, 3, 4, 5, 6, more than two molecules are present at the outer side. So that's why this molecule can directly pass from the higher concentration to the lower concentration. And this is called as the along the concentration gradient. Okay. So the passive transport, it is two types of the passive transport. One is called as the simple diffusion. Simple diffusion. Another is the facilitated diffusion. What is the simple diffusion? The simple diffusion means the gases, for example, the oxygen and the carbon dioxide, they can pass easily through the plasma membrane along the concentration gradient. Let's say this is the cell. In the inside, let's say the carbon dioxide is more. Suppose in the outside, the carbon dioxide is a low concentration. That means, let's suppose the 5 molecules of the carbohydrate is present at the inside and 3 carbo CO2 molecules are present at the outside. That means in the inside more carbohydrate is present. So more carbon dioxide is present. That means that now the carbon dioxide transports from the higher concentration to the lower concentration or the along concentration gradient. That means from the inside to the outside. Let's suppose in the inside the oxygen is present in the lower concentration. At the outside the oxygen is present at the higher concentration at the outside. Now the oxygen comes from the outer to lower. That means molecule which is present higher concentration and the movement is always from the higher to lower concentration. If it is present at the higher in the inner surface, then it transports from the inner to higher. If it is present higher in the outer, then it transports from the higher to the outer to the lower, outer to the inner layer. So this is the simple diffusion. One is the gases, another is the non-polar molecule. For example, benzene. Benzene is a non-polar molecule. Why the non-polar molecule they can pass? Because the lipid layer is itself it is a non-polar or the hydrophobic. Always the lipid bilayer, the plasma membrane is hydrophobic. So that's why the hydrophobic molecule they can easily pass the layer because it can means this is the hydrophobic molecule. Here it is also the hydrophobic molecules. That's why they can mix each other and they can easily pass through the hydrophobic layer. But if it is a hydrophilic molecule, this is the hydrophilic molecule. The hydrophilic molecule, they cannot pass through the hydrophobic. Why? Because the hydrophilic cannot interact with the hydrophobic molecule. Because the hydrophilic means those molecules which interact with the water molecule. The hydrophobic means those molecules which always wants to away from the water molecule. That's why the hydrophilic cannot interact with the hydrophobic molecule. That's why the hydrophobic molecule cannot pass. But the hydrophobic molecule, they can pass through the layer. So that's why gases, oxygen, carbon dioxide, any gases, and the non-polar molecule, that means the hydrophobic molecule, they can pass also the plasma membrane from the higher concentration to the lower concentration. Another most important thing, one is called as the diffusion. What is the diffusion? Diffusion is the transport of water molecule through the simple diffusion. What is the not diffusion? That is the osmosis. Osmosis. What is the osmosis? The osmosis means the transport of the water molecule through the simple diffusion. That is called the osmosis. So that means the osmosis is the sub part of the simple diffusion. That means the simple diffusion, the gases can be passes from the higher concentration to the lower concentration. The non-polar molecule, they can also passes from the higher concentration to the lower concentration. And the water molecule also can passes from the higher concentration to the lower concentration through the simple diffusion. And this process is called as the osmosis. So what is the osmosis? It is the movement of the water through the simple diffusion. Through the simple diffusion is called as the osmosis. Okay, so this is the simple diffusion. That means it always moves from the higher to lower concentration or along the concentration gradient. That's why, because it transports from the higher to lower concentration, it does not need any energy. It does not need any energy. So another is the facilitated diffusion. So what is the facilitated diffusion? For example, some molecule, let's say protein. Let's say this molecule, it is the protein. Normally, the protein is the hydrophilic molecule. Hydrophilic molecule. Let's say this protein has higher concentration at the outer side. Let's say this is the protein molecule, and it is the hydrophilic. Okay, it is present at the higher concentration at the outer side, and the lower concentration, higher concentration at the outer side, lower concentration at the inner side. But it does not pass through the membrane directly, because this is the hydrophilic molecule. It cannot interact with the hydrophobic core group, because the plasma membrane of the lipid is the hydrophobic group. But if the non-polar molecule, non-polar, for example the benzene, if it is present at the higher concentration at the outer side, lower at the inner side, it can directly pass through the layer because the lipid layer is also the hydrophobic, the non-polar, the benzene is also the hydrophobic. So it can interact with each other and they can easily pass through the membrane. But now the protein is hydrophilic. It does not, or it cannot pass through membrane directly. So that's why it needs some assistant, and this assistant is called as the carrier. That means it needs some carrier for the transport of the molecule from the higher to lower. So this is called as the facilitated diffusion. That means some facilitated needs for the transport of this molecule, and this is called as the carrier, or it is called as the carrier mediated facilitated diffusion. This is called as the carrier mediated facilitated diffusion. Okay, so now there are two types of the transport. One is called as the simple transport, another is the facilitated diffusion, another is the simple diffusion. So what is the simple diffusion? So the gases, oxygen, carbon dioxide, non-polar molecule like the benzene along with the water. Water is the exception, that means it transports through the process that is called as the osmosis. So osmosis is simply the simple diffusion in which the molecule is only the water. That means the movement of the water through the simple diffusion is called as the osmosis, and they along with the facilitated diffusion. The facilitated means the hydrophilic molecule or some polar molecule, if it is present higher concentration, then it transports from the higher to lower concentration, but it is facilitated by some carrier, that is called as the carrier mediated facilitated diffusion. Okay, so this is the simple and the facilitated diffusion. So now come to three types of structure. One is called the hypotonic, isotonic, hypertonic. Hypotonic means it is the inside or the inner side, this is the outside. Hypo means low, hyper means high. So in the solution, one is called as the solute, another is called as the solvent. When the solute means the hypotonic, isotonic, hypertonic solution, they are named according to the solute concentration. That means in the hypotonic solution, at the outer side, more solute concentration, so means low solute concentration is present. That means in the inside, higher solute concentration is present. At the outer side, lower solute concentration is present. In the outer, low solute. So if the low solute is present at the outer side, that means more solvent is present. In the inside, higher solute is present, that means low solvent is present. Isotonic means in the outer side and the inside all are same. This is called the ideal condition of the isotonic solution. This is the hypertonic solution. In the hypertonic solution, the outer condition contains more solute at the outer. That means low solvent is present at the outer. Now in the exam it will ask that if any cell, let's say this is the solution and this is called as the hypertonic solution. Hypertonic solution. In the exam if it will ask that, let's say this is the one molecule or the one cell, if one cell is transported or is placed to the hypertonic solution, then what will be the condition of that cell? Then the answer will be shrink. Why? Let's say in this area. Now the cell is placed in the cell, inside the cell. This is the hypertonic solution. This solution, this is called as the hypersonic solution. In the hypersonic solution, the solute concentration, the solute is more and the solvent is less, like this. So that means in the inside, the solute will be low and the solvent will be high. So that means let's say the solvent is the water. The water molecule is present. This is the solvent, means water. So now that means the water molecule is present higher concentration in the inside and the lower concentration is the outer side. So that means it transports from the higher to lower. That means the water released from the higher to lower. That means from the inner to higher. So that means after the transport of the cell, the placing of the cell to the hypertonic solution, after the transferring the water molecule from the inner to higher, it will shrink like this. Why? Because after transporting the hypertonic solution, the solution of the water concentration is less at the outer side and the water concentration is more in the inside. That means water, they transport from the inner to lower. That means from the cell to the outer solution. So that's why it will be shrink. In the exam, if it will be asked that if the cell is placed in the hypertonic solution, it will shrink. So like that, if the cell is transported to the hypotonic solution, let's say this is the cell, this is the hypotonic solution. In the hypotonic solution, the solute concentration is less and the solvent is more. That means in the inside the solvent is less. Solvent means less than water. Obviously in the cell the solvent is the water. That means in the hypotonic condition the water molecule concentration higher in the outer side and then go lower in the inside. So from the higher to lower, that means the water, they transports from the higher solution, from the outer solution inside to the inside the cell. That means from this solution, it transports to the cell, so that after transferring so many water molecule, the cell will be burst. If the many water molecule, they are transported inside the cell, it will be burst. So if the cell is transported to the hypertonic solution, the cell will be shrink. If it is transported to the hypotonic solution, it will be burst. Okay. So now come to this is the passive transport. Okay, passive means one is the simple diffusion, another is the facilitated diffusion. Simple means the gases, the non-polar and the water molecule, they can pass easily from the higher to lower concentration or along the concentration gradient. Facilitated means the polar molecule transport from the higher to lower concentration. But what if, let's say, this is the cell. Always inside the cell, the sodium concentration is very low, and the outer side, the sodium concentration is high. And always inside the cell, in any cell, the potassium concentration is high, and in the outer surface, the sodium concentration the sodium concentration is low. Okay. So that means in the simple diffusion or the facilitated diffusion, it always the sodium transports from the higher to lower. That means from the outer to the lower, from the outer to the inside. The sodium transports, but the potassium through the passive transport always transports from the inside to the outer side. Okay. But in sometimes the potassium transports from the outer to lower, or the sodium transports from the lower inside to the outer side. And this type of the transport is called as the active transport. Active transport. So that means here the active transport means transport of the solute or the transport of the molecule against concentration gradient. Against concentration gradient means from the lower concentration to the higher concentration. So because against concentration gradient, it needs some use of the energy. Look at this structure. In this structure, let's say this is the outer, this is the inner layer. In the outer layer, low concentration is present, and the inside higher concentration is present. So if in the passive transport, it can easily pass from the inner to the outer, but it cannot pass from the outer to the lower. But if it wants to transport from the outer to the lower, or from the lower concentration to the higher concentration, it needs some energy. Here the energy is the ATP. Don't confuse between the energy and the ATP. That means if it will be asked that in the active transport, the energy is needed or ATP is needed, always mention the energy is needed. Why? Because ATP is one of the type of the energy. That means we use ATP as a form of the energy. For example, some bacteria like halobacteria, they use light as a source of energy here. But still it is called as active transport because of the investment of because of the use of the energy, not the ATP. We use ATP as a source of energy. Halobacteria, they use light as a source of energy. So that means in the active transport or the transport which moves against the concentration gradient, that means from the lower to higher concentration, the energy is needed. Either it may be in form of the ATP or it may be in the form of the light. Okay. So one of the most example is the sodium potassium pump. Sodium potassium pump. Okay. Now this is the sodium potassium pump. This is the sodium potassium pump. This is the extracellular fluid. That means it is the outer side. This is the cytoplasm. That means it is the inner cell. So I have already told that always inside the sodium concentration is lower and the potassium is higher. On the outer side, the sodium concentration is higher and the potassium concentration is lower. So that means in the passive transport, potassium can easily pass from the inner to outer and the sodium can easily pass from the outer to the inside. But if sodium wants to come from inner to outer, that means it is the against. Or the potassium, it wants from the outer to inner. It transports, this type of the transport is called as the active transport, and this type of the is called as the sodium potassium pump. Look at this structure. This is called as the, this type of the structure, it is called as the sodium potassium pump. And here, three sodium, 3 sodium molecules are transported from the inner to the outer. And like that, 2 potassium molecule transports from the outer to the inner. And this is called as the active transport, and here the ATP is needed as a source of the energy. So the sodium potassium pump, it is the example of the active transport through which 3 sodium and the 2 potassium are transported at the same time through this pump, and this pump is called as the sodium potassium pump. Okay, so this is the all about the plasma structure and the transport of the across the plasma membrane. Okay, if you like it, then please subscribe. Thank you. Hello everyone. Today we will know about the endomembrane system. So what is the endomembrane system? So endo means inside and membrane means of course the membrane. So endomembrane system, that means the system that is present inside the membrane. So of course membrane means it is the plasma membrane. That means a system is present inside the plasma membrane is called as the endomembrane system. But before going to the endomembrane system, we should know something about the cell. So this is the cell, and the outer layer is called as the plasma membrane. And inside the cell, of course, the cytosol, the white part, are called as the cytosol, or the liquid part or the watery part is called as the cytosol. And many organelles are present. For example, this is the nucleus. This is the nucleus, and inside the nucleus, a round structure like this is present, and this is called as the nucleolus, and this is called as the mitochondria, and this is called as the peroxisome, and this is the ribosome that is present in the endoplasmic reticulum, and this is the vacuole, and this is the lysosome, this is the Golgi complex. So the cell organelles can be two types. One is called as the single membrane bound organelle. Single membrane bound organelle, and another is called as the double membrane bound organelle. So what is the single membrane bound organelle? What is the double membrane bound organelle? For example, if we look the vacuoles, so here the vacuoles, here the membrane is one. That means this organelle or the vacuole is surrounded by a single membrane or single layer, that is called as the single membrane bound organelle. For example, if we look the mitochondria looks like this. This is a one layer, and inside that layer, this structure is like this. That is called as the cristae. So that means in the mitochondria there are two types of two membranes are present, and this is called as the outer membrane, and this is called as the inner membrane. So that means all the enzymes, all the materials that are required for the mitochondria, that is present inside the cristae or inside the inner membrane. So that means the mitochondria is a double membrane bound organelle. So that means the mitochondria has two types of two membrane. One is called as the outer membrane, and another is called as the inner membrane. For example, the nucleus. Look at the nucleus structure. Here it has a two layer. This is the one layer, and inside it another layer is present. So the outer layer, of course, it is called as the outer membrane, and the inner membrane is called as the inner layer or inner membrane. So that's why the mitochondria, nucleus, and another is the chloroplast. So it is also a double membrane bound organelle, because the mitochondria, nucleus, and the chloroplast, they have two membrane. That means the boundary has two. They have two boundaries. That's why it's called as a double membrane bound organelle. So all other organelles are called as a single membrane bound organelle. For example, ER or the endoplasmic reticulum, the Golgi body, lysosome, a vacuole, and all other organelles except the mitochondria, nucleus, and the chloroplast, they all are the single membrane bound organelle. So now come to what is the endomembrane system. So endo means inside, the membrane means the membrane. That means a system is present inside the membrane, that is called as the endomembrane. So what is the system? So that means a system is present or it is responsible for the formation or production, modification, packaging, and transport. So that means a system is present inside the membrane which are responsible for the formation, modification, packaging, transport of protein and lipid. So that means it is a system. That means a system is coordinated to form or to formation, modification, packaging, transport of protein and lipid. So what are the system? The system includes endoplasmic reticulum, Golgi body or Golgi complex or Golgi apparatus, and lysosome and vacuoles. So that means these four organelles are responsible for the formation, modification, packaging and transport of protein and the lipid. Look at the structure. This is the ER. This structure is called as the ER. So ER, after that it is the Golgi complex, then it is the lysosome, and then it is the vacuole. So that means these four organelles are responsible for the protein and the lipid formation, modification, packaging and transport. That's why the endomembrane system includes these four organelles. Why these organelles? Because these four organelles are cordially managed to formation of this protein and the lipid and transport, of course. And other organelles, for example, mitochondria, chloroplast, and peroxisome. So they are also present, but they have different function. That means they have their own function. For example, this mitochondria, they have it have its own function. So that means they don't include in the endomembrane system. So endomembrane system includes the endoplasmic reticulum, Golgi, lysosome and the vacuoles. So if we look this ER and the Golgi, if we zoom it, it looks like this. So that means if I zoom it, it looks like this type of the structure. So now come to the endoplasmic reticulum. Why it is called as the endoplasmic reticulum? Endoplasmic reticulum. So endo means inside, plasmic means cytoplasm. So reticulum means it's a network. So that means inside the cytoplasm, a network-like structure is present, and that is called as the endoplasmic reticulum. Look at the structure of the endoplasmic reticulum. It is like a network-like structure. So that's why it is called as the endoplasmic reticulum, because it is present inside the cytoplasm. So the endoplasmic reticulum, it was first seen in the electron microscope by the Garnier. But if in the exam, in the NEET exam, it will be asked that who coined the term ER, and the answer will be Porter. So Porter coined the term ER, or the endoplasmic reticulum. So the endoplasmic reticulum, it is found in all the cell except mature RBC and embryonic cell and prokaryotic cell. So that means, of course, the prokaryotic cell, they don't have the ER, and the eukaryotic cell, the mature RBC and the embryonic cell, they also don't have the endoplasmic reticulum. So now come to the structure of the ER. If we look the structure of the ER, the structure look like, there are two types of the structure. One is called as the cisternae and another is called as the tubule. So what is the cisternae, what is the tubule? Look at this structure. This flattened-like structure is called as the cisternae. So they are the cisternae. So what is the cisternae? The cisternae is the long flattened unit. Long flattened unit, and they are the hollow. So hollow means it looks like the pipe. In the pipe, it looks like this. That's the pipe. That means inside the pipe there is the hollow part. So the cistern look like this, but it is a branch. It's not a straight. Like it is, the cistern look like this. So that means a pipe-like structure or flattened-like structure of ear is called as the cisternae, and another structure is called as the tubule. So what is the tubule? The tubule means as the name suggests, it is the tube-like structure. It looks like this. Here also, it is a tube-like structure. So they are called as the tubule. So that means inside the cisternae and the tubule, inside the hollow part. That means inside like the pipe, inside there is a hollow part in the cisternae as well as in the tubules. So in the ear part, there are the classified again the ear as luminal, and another is the extraluminal. So what is the luminal? What is the extraluminal? So luminal means inside. So extraluminal means cytosolic. So what is the inside? For example, some molecule is present here. So that means it is present inside the ear. So that means we can say that this molecule is present luminally. That means this molecule is present inside the endoplasmic reticulum. So what is the extraluminal? That means cytosolic. Simply it is the outside of ear. So let's suppose the molecule is present here. That means it is present outside the ear. So now we can say that the molecule is present extraluminally. That means the molecule is present in the cytosol or in the outside of the ear. So this is the luminal and the extraluminal part of the ear. So now come to the major difference between major structure of ear. The ear can be classified, or ear can be divided into two parts. One is called as the rear and another is called as the cere. The rear is called as the rough endoplasmic reticulum, and cere is called as the smooth endoplasmic reticulum. So what is the rough endoplasmic reticulum, and why it is called as the rough, why it is called as the smooth? Look at the structure here, from here to here. So in this structure, a blue color structure is like this, they are present, and these are called as the ribosome. So that means in the rough endoplasmic reticulum, the ribosomes are present. So because of the presence of the ribosome, it is rough. While in the smooth endoplasmic reticulum, look at this structure. So in this structure, there is no ribosome. So that means that's why it is called as the smooth endoplasmic reticulum, or it is called as the cere. So this structure is called as the rough endoplasmic reticulum because of the presence of the ribosome on the membrane of the rough endoplasmic reticulum. So if you look the endoplasmic structure, it emerges from the nucleus. This is the nucleus, this is the ear, and this is the Golgi. So look at the structure. The ear emerges from the nucleus part, and that too nucleus has, we have already discussed that nucleus is the double membrane bound organelle. That means the nucleus has two layers. one is called as, this is called as the outer layer, and this is called as the inner layer. So that means the ear emerges from the inside or the inner layer of the nucleus. That means, let's say this is the first nucleus, and from this the ear emerges like this. Let's say this is the nucleus. The ear emerges from the inner membrane of the nucleus. So this is called as the rough endoplasmic reticulum because of the presence of the ribosome, and the smooth because they don't have the ribosome. So this is the main difference between the rough and the smooth endoplasmic reticulum. So why the ribosomes are present? Actually, in the ribosome, in the rough endoplasmic reticulum, there is a protein is present that is called as the ribofourin. Ribofourin, that is a protein present on the rough endoplasmic reticulum, and that helps in the attachment of the ribosome. That means it's simply, you can say that it's a like glue-like structure. That means this protein which helps the attachment of the ribosome in the rough endoplasmic reticulum. So now come to what is the function of the rough endoplasmic reticulum. That means what does it do? So the rough endoplasmic reticulum, it helps in the protein synthesis. Protein synthesis. That means in the rough endoplasmic reticulum, it helps in the protein synthesis. Please look at this term. It helps in the protein synthesis. In the NEET exam, there are the two types of the question it will be asked. That if the question is which organelle helps in the protein synthesis, the answer will be the ear. Now the second question is, what is the site of protein synthesis? So that means, what is the site of protein synthesis? That means on where the protein synthesis is occurred. So the answer will be not endoplasmic reticulum. The answer will be the cytoplasm. So that means in the rough endoplasmic reticulum, it helps in the protein synthesis. It synthesizes the protein, but the protein synthesizes exactly in the cytoplasm like this structure. So once the protein is synthesized in the cytosol, now it enters inside the rough endoplasmic reticulum. But first it synthesizes in the cytoplasm. Please remember this. If in the NEET exam it will be asked that what is the site of protein synthesis, the answer will be cytoplasm. But of course the rough endoplasmic reticulum helps in the protein synthesis. That means rough endoplasmic reticulum with ribosome, it helps in the protein synthesis, but protein synthesis occurs exactly in the cytoplasm, and after the formation, then it enters inside the endoplasmic reticulum. So this is the main function of the rough endoplasmic reticulum. And which type of cell contains the rough endoplasmic reticulum? Mainly liver, pancreas, and fibroblast. So that means the liver, of course, it contains many enzymes, many proteins, many lipids. So because of this, means liver is responsible for the formation of many enzymes or many proteins. So that's why liver contains high amount of the endoplasmic reticulum. Along that, pancreas is also site of the production of the many proteins and the lipids. And the fibroblast means it's simply a connective tissue. It synthesizes like the collagen. Collagen is synthesized by the fibroblast. So the fibroblast was also contains the endoplasmic reticulum. So now come to again first rough endoplasmic reticulum. It synthesizes the protein at the outside. Let's say this is the protein and it is protein. This protein is synthesized at the outside. Now after that it enters inside the endoplasmic reticulum. So that means the first it is the extraluminal, and after the production it becomes the luminal or inside the endoplasmic reticulum, and after that it forms a vesicle-like structure. Vesicle means simply a structure which is surrounded by a membrane, and inside the vesicle the protein is transported. And now this protein, this is a protein, so now this protein enters into the Golgi. This is the main function of the rough endoplasmic reticulum. That means it is helps in the protein synthesis, and after protein synthesis it transports to the Golgi by forming a vesicle-like structure. Vesicle-like structure. Okay, this is the rough endoplasmic reticulum. Rough endoplasmic reticulum. These all are the rough endoplasmic reticulum because of the presence of the ribosome on its membrane. So another is called as the smooth endoplasmic reticulum. So why it is smooth? Of course, it is they don't have the ribosome, and they have only the tubule-like structure. Look at the structure. Here it is called as the smooth endoplasmic reticulum. Only the tube-like structure is present. That means only the tubules are present in the smooth endoplasmic reticulum. So what is the main function of the smooth endoplasmic reticulum? So it is the site of lipid synthesis, lipid as well as steroid, lipid and steroid synthesis. So that means the rough endoplasmic reticulum, it helps in the protein synthesis, and the smooth endoplasmic reticulum, this helps in the lipid synthesis or lipid and the steroid. So now which cells contain the smooth endoplasmic reticulum? The cells like liver, a fat cell, and adrenal cortical cell. So adrenal cortical cell you can simply call as adrenal cortex. So what is the adrenal cortex? You will know about the hormone part, that in the adrenal there are the two parts, the two layers. One is called the inner and it is called as the medulla, and the outer is called as the cortex. In the cortex part, it synthesizes three hormones. One is called as the glucocorticoid, mineral corticoid, and another is the androgen. So these all are the steroids. So that means since the smooth endoplasmic reticulum, it is responsible for the lipid and the steroid synthesis. That means the liver, of course, it also contains, it also helps in the, it is means responsible for the production of protein and the lipid also. So like that fat cell, of course, it synthesizes the lipid, and the adrenal cortical cell, that means the glucocorticoid, mineral corticoid, androgen, they all are synthesized by the cortex. So that means the liver, fat cell and the adrenal cortex cell, they contain the smooth endoplasmic reticulum. Look at the structure. Here also, let's say this is the lipid is present. Here also, like the rough endoplasmic reticulum, it also forms a vesicle-like structure. And inside it, a lipid, let's say it is the lipid. Inside it, the lipid is present, and now the lipid is transported again into the Golgi. So what is the function of rough endoplasmic reticulum and the smooth endoplasmic reticulum? Rough endoplasmic reticulum function is the production of the rough protein, and after that it forms the vesicle and transports to the Golgi. Smooth endoplasmic reticulum, it synthesizes the lipid and the steroid, and then it forms the vesicle-like structure and it's transported to the Golgi again. So another main function of the smooth endoplasmic reticulum is it helps in the detoxification. It is a neat question. Many times it will be asked that. So which organelle helps in the detoxification? The answer will be endoplasmic reticulum. But if in the option that is called as the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi, lysosome, then answer will be smooth endoplasmic reticulum. The smooth endoplasmic reticulum has one more extra function is it helps in the detoxification. And detoxification is occurs in the which organelle? It is the liver. Maximum detoxification occurs in the liver. That's why liver also contains high level of smooth endoplasmic reticulum. Okay. So this is all about the rough endoplasmic reticulum. So now come to the Golgi body. It is called as the Golgi body, or it is called as the Golgi apparatus. So, or it is called as a Golgi complex. You can say whatever you can say, that Golgi body or Golgi apparatus or Golgi complex. So this is the structure of the Golgi. So the Golgi, it was discovered by Camillo Golgi in 1898. So the first electron microscopic structure of the end of Golgi body look like this. That means it is a densely colored structure that is found in the electron microscope. Look at the structure, and this is the Golgi body, and it was seen in the electron microscope for the first time, and it was densely, means a very black color or very darkly stained the color of the Golgi body was seen. So that's why it is called the densely colored structure that was present inside the cell is called as the Golgi apparatus. So the Golgi apparatus is found in all the cell except mature RBC, sieve tube, and sperm of bryophyte and pteridophyte. Okay, so that means mature RBC, sieve tube, and the sperm of bryophyte and the pteridophyte, they don't have the endoplasmic reticulum. So in the animal cell, 3 to 7 complex are present. 3 to 7 means this is the 1, this is 2, this is 3, this is 4. That means 3 to 7 complex structure of the Golgi are present, but it is found very large in size in case of the animal. But in the plant, the number is 10 to 20, but the size is very, very small. So that means the animal, the number is huge, but number is very low, but the size is very huge. But in the plant, the number is very huge, but the size is very small. And that's why in the plant, the Golgi body is called as the dictyosome. Dictyosome. So that means what is the dictyosome? It is simply the Golgi body that is present inside the plant is called as the dictyosome. So come to the structure of Golgi body. This is the structure of the Golgi body. The structure contains three types of the structure are present. One is called as the cisternae. One is called as the tubule. And another is called as the vesicle. So what is the cisternae, what is the tubule, what is the vesicle? The cisternae means it's like this flat-like structure. They are called as the cisternae. The flat, long, disc-like shape, flat, disc-like shape is called as the cisternae, and its the size is 0.5 to 1 micrometer. What is the micrometer? Simply the 10 to the power minus 6 meter is equal to 1 micrometer. So that means 1 meter by 10 to the power 6 is 1 micrometer. So this is the diameter of the cistern. So that is another is called the tubule. What is the tubule? And at the periphery there are many structure looks like this. And this is called as the tubule. And another is called as the vesicle. Vesicle, that means this is a vesicle. So that means a structure in which it is surrounded by a layer and it contains something that is called as the vesicle. So there are three types of the structure. One is the cisternae, a long-like structure. The tubules, a very small that is present at the periphery is called as the tubule, and the vesicle, that means from the Golgi, the vesicle arises and it contains some food particle. We will know something after this Golgi body study. We will know about what the vesicle. So now come to the cisternae. It is very, very important. Cisternae means, look at the structure. This is the nucleus, this is the ear, and this is the Golgi. The Golgi part, it has two parts. One is called as the convex and another is called as the concave. Means, let's say this is the nucleus, this is the ear, and the Golgi is present, looks like this. Look at the structure. That means one part is called as the convex and one part is called as the concave. You know what is the convex, what is the concave. The pointed end is called as the convex. So that means one part is here, this part is called as the convex and this part is called as the concave. So the convex part is also called as the cis face, while the concave part is also called as the trans face. That means here it is called as the cis, here it is the trans. That means here this part is called as the cis part, and here this all the large structure they are called as the trans part. Or it is called as the forming phase. Forming phase, and it is called as the mature phase. Why it is called as the forming, why it is called as the mature? Means if in the child the size it looks like this, but after the adult, of course the size increases. So that means it is the forming phase, it is the mature phase. Here also look like this structure. That means it is a forming phase. That means it is just formed here, but it is the already matured one. So that's why the trans part is called as the mature phase. And always remember that this C phase is facing towards nucleus. Towards nucleus. Look at the structure. This phase always looking towards the nucleus. That means it is nearly present at the nucleus in comparison to the trans phase. So that means this phase is very far in comparison to this C phase. So this is the C phase, this is the trans phase. The C phase is the convex, the trans is the concave, and the C is forming phase, and the trans is mature phase, and the C is towards the nucleus, and the trans is away from nucleus. Okay, so they are interconnected. That means the Cs and the trans, they are interconnected here. So this is the main structure of the Golgi body. So now come to what is the function of the Golgi body. So that means in the ear, from the ear, the proteins are transferred by forming a vesicle. Here in the smooth endoplasmic reticulum, the lipids. The lipid, here it is the protein. So they are synthesized and transported to the Golgi. So in the Golgi, the C phase accept the protein and the lipid. So always important, the C phase accept the protein and the lipid and the steroid. Okay. So what is the function of the lipid? Function of the Golgi. That means after accepting, what will it do? So look, let's say this is the protein. Now the protein comes inside the Golgi. Now the Golgi forms the modification of this protein. That means it helps in the modification of protein and lipid. That means let's say this protein, this enters into like this, here in the lipid, here also it enters like this. So after this it modifies this protein and the lipid. So what is the modification? The Golgi body adds a carbohydrate into him. So the process is called as the glycosylation. So what is the glycosylation? That means the addition of the carbohydrate to any molecule. So here in the protein and the lipid, the main function of the Golgi is it adds a structure here also, it is called as the carbohydrate. That means it modifies the protein and the lipid by adding the carbohydrate, and the process is called as the glycosylation. The second is called as the most important protein sorting. The cell sorting. Sorting means, let's suppose this is a cell, this is a nucleus, this is ER, and this is the Golgi. So that means in the ER, the protein and in the smooth endoplasmic reticulum, the lipid are present, lipid are transported into the Golgi, and after that the glycosylation occurs, and after that it decides this protein or the lipid should go to where. That means let's say there are many organelles, for example the mitochondria and lysosome, and many organelles are present. Let's suppose this protein is for the mitochondria. That means the Golgi decides whether it should go to the mitochondria or not. If it decides it goes to mitochondria, then this structure, that means after the glycosylation, the structure, it also forms the vesicle like this structure. Here, this is the vesicle. The vesicle-like structure, now it enters into the mitochondria. Let's suppose it decides this protein or the lipid should go to the membrane. Then it forms again the vesicle, and this vesicle fuses to the plasma membrane. Looks like this. This is the protein or the lipid that is needed for the plasma membrane. So it fuses like this. Let's suppose it decides this protein or the lipid should go to the lysosome. So again it forms the vesicle. So now this vesicle it fuses to the lysosome, and now lysosome should get its protein or the lipid. So this is called as the protein setting. That means it decides which protein or the lipid should go to the which organelles. So that's why the Golgi body is also called as the traffic police of the cell. So that means in the traffic police, like that, the traffic police decides which side of the people should go to where. So like that, the Golgi also decides which molecule or the protein should go to where. So this is the main function of the Golgi, and along with that, it helps in the formation of cell wall by dictyosome. So that means in the plant the Golgi is called as the dictyosome, and the Golgi of the plant or the dictyosome helps in the formation of the cell wall in the plant cell. And another is it helps in the acrosome formation in sperm. So what is the acrosome formation? That means during the fertilization, let's say this is the ova, this is the sperm. So it is some reaction is required for the fertilization, and this reaction is called as the acrosome formation. So the Golgi in the sperm it also helps in the acrosome formation that is needed for the fertilization. So this is the main function of the Golgi. That means accepts the protein and the lipid, and after accepting it modifies by adding the carbohydrate, or the process is called as the glycosylation. Then the cell sorting, that means it decides which protein or the lipid should go to where. The next one is it helps in the formation of the cell wall by the dictyosome, and another is it helps in the acrosome formation in the sperm. Okay, so this is all about the Golgi apparatus. So now come to the another structure that is called as the lysosome. Okay, the lysosome, it was discovered by Christian D. Dubé in 1955. So it is, of course, it is a single membrane bound organelle. That means it is bound by a single layer, and it is formed by the process of the packaging in the Golgi apparatus. For example, look at the structure. This is the ER. So this is the Golgi, and this is the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum, it synthesizes the lipid and it transports to the lipid, it transports to the Golgi. And here also the smooth rough endoplasmic reticulum, it synthesizes the protein and transports into the Golgi. And after that, the Golgi apparatus sorts, that means the protein and the lipid, it should be decided by the Golgi apparatus. So now come to the lysosome. So like the packaging, the lysosome, this is the lysosome. So that means the lysosome is also formed by the process of the packaging of the Golgi body. So what is the function of the lysosome? That means here also this lysosome is also formed by this Golgi body. So what is the lysosome? It's simply, let's say this is the lysosome. It contains many hydrolytic enzymes. Hydrolytic enzyme. So what is the hydrolytic enzyme? That means it is a digestive enzyme or simply acidic enzyme. So that's why what is the pH of lysosome? Always it is the very acidic, because the hydrolytic enzymes are present or the digestive enzyme. For example, protease. Protease, that means it digests the protein. Carbohydrate, that means it helps in the digestion of the carbohydrate. So nuclease, that means it helps in the digestion of the nucleic acid or the DNA and RNA. For example, the lipase. So lipase means it helps in the degradation of lipid. So that means all these hydrolytic enzymes or all these digestive enzymes are present inside the lysosome. So first, next come to the what main important is the polymorphism. Polymorphism of lysosome. So what is the polymorphism? That means more than one form of lysosome. That means the lysosome of different types of the lysosome are present. So that is called the polymorphism. So it is also a neat question. That means which organelle shows the polymorphism? The answer will be lysosome. So the lysosome are the three types of the lysosome are present. One is called as the primary lysosome, another is called as the secondary lysosome, and another is called as the autophagosome. The secondary, it is also called as the heterophagosome. The primary is simply called as the primary. So what is the primary lysosome? That means in this primary lysosome, here also the hydrolytic enzymes are present, but the hydrolytic enzymes are present in the inactive condition. Inactive means here also the enzymes are present, but it is not active. For example, in this structure also, let's say any protein comes to this, it cannot digest because the protease is present, but the protease is present in the inactive condition. So that means in the primary lysosome, the proteins are present, but it is present in the inactive condition. But in the secondary or the heterophagosome, the protein is present and that too in the active condition. So that means here all the molecule, that means protein, carbohydrate, lipid, nucleic acid, phosphate, all these things, they are digested by this secondary lysosome. So that's why the secondary lysosome, the main function is the digestion of the food. So what is the digestion of the food? Simply, let's say in the stomach. Let's say in the stomach. In the stomach cell, if I cross-check it, there are many different types of the cells are present. So if in the exam it will be asked that which type of the cell, or in the cell, let's say this is the one cell of the stomach cell. So in the cell of the stomach, which organelle digest the in the stomach, the proteins are digested. That means in the cell of the stomach, which organelle digest the protein? The answer will be the lysosome. So that means the lysosome of the stomach digests the protein. So that means in the liver also, in the small intestine also. So that means they all contains the lysosome. That means inside in these liver, small intestine, and in the stomach. That means all the alimentary canal. That cell contains the nucleus, the lysosome more vigorously, because this lysosome helps in the digestion of the food. Another important is that lysosome which also helps in the digestion of foreign particle. What is foreign particle? That is simply called as the antigen. For example, let's say this is a bacteria or the virus or the bacteria. So what is the foreign particle? That means anything which is not about the cell or not about our body, I mean outside body material. Let's say this is a bacteria, it enters into it. So now this bacteria, the lysosome digests the bacteria because the lysosome contains the acidic enzymes, the hydrolytic enzyme. That means this hydrolytic enzyme can digest anything. So it also digests the bacteria also. So that is the main function of the lysosome. One is the digestion of the food. Along with that, it helps in the digestion of the foreign material, the foreign particles such as bacteria, virus, pollen. What is the antigen? Different types of the antigen, they can be digested in the lysosome. And another is the autophagosome. Autophagosome means, let's suppose this is the nucleus, this is the ER, and this is the Golgi. So this is the lysosome. This is the, let's say, mitochondria. So if the mitochondria is damaged, so now that means it is now not in use in the cell. So this mitochondria, it fuses into it and it enters into the lysosome, and lysosome digests its own cell because it is not now the use of the cell. So this autophagosome, it digests its own organelle which are damaged or dead, of course. That means it is not use of this. So that means the autophagosome means digestion of its own organelles. So that's why the lysosome is also called as the suicidal bag of cell. It is very, very important. It is many times asked in the NEET exam that which cell organelle is suicidal bag of the cell, and the answer will be the lysosome, and that too autophagosome one. Okay. So this is the all about the lysosome structure of the cell. So now come to the vacuole. So what is the vacuole? Vacuole is also a membrane-bound organelle, and that too single membrane-bound organelle. Okay. So this is the, let's say this is the plant cell, and this is the vacuole, the blue color hole, the part, it is called as the vacuole. And the plasma membrane of the vacuole is called as the tonoplast. So what is the tonoplast? The tonoplast is simply the plasma membrane of vacuole. So what is the role of this tonoplast? Okay, before going to that, what is the role of the vacuole? So the vacuole contains water, sap and the excretory product. So what is the sap? Sap means simply any liquid part that is present in the plant is called as the sap. So water, sap and the excretory product, and the vacuole contains this type of material, which is interestingly not useful for the cell. Not useful for cell. Okay, so vacuole contains this water, sap, excretory product that is not useful for the cell, and the vacuole are very, very large. That means, look at the structure. This is the mitochondria, looks like very teeny. This is the nucleus, it is also very teeny. But look at the vacuole, the structure. That means in the plant, 90% of total volume is occupied by the vacuole in the plant cell, of course. That means the vacuole is so much large that it enters or it occupies 90% of total volume of the plant cell. But in the animal cell, normally in the animal cell, it can be found or it can be absent. So that means if it is found, it is present in the very less in number and very, very small, very, very small. That means in the plant cell, the vacuole looks like this. In the animal cell, the vacuole looks like this. But in the plant cell, this is a very huge, this vacuole. So what is the main function of the vacuole? The vacuole main function is this is the tonoplast, and through the tonoplast, the ions of the solute are transported into it through active transport. So what is the active transport? In the previous video, we have studied about the active transport. That means it uses the energy and the solute, it transfers from low concentration to high concentration. So that means here the ion concentration, let's say this is the ion. For example, sodium, potassium. The ion concentration is very low, and inside the vacuole the ion concentration is very high. And this solute or these ions are transported into this by this membrane, and this membrane is called as the tonoplast. So through the membrane of the tonoplast, the ions are transferred by the active transport. That means by the use of the energy. That means from the lower concentration to the higher concentration. So in the NEET exam, it will be asked that what is the concentration of the ion inside the vacuole? Always it is very, very high, because it always transports the ions inside it by the active transport. Okay. So now the vacuoles are mainly two types. Vacuoles are mainly two types. One is called as the food vacuole, and another is called as the contractile vacuole. So what is the food vacuole? Food vacuole means simply it digests the food like the lysosome, but less than 1%. That means maximum digestion of the food occurs in the lysosome, and very small amount of the food is digested in the vacuole. And another is that it found in protozoan, protist, and lower animals. What is the function of the contractile vacuole? Look at the amoeba. Let's say this is the amoeba. So in the amoeba, here is the contractile vacuole. That means it regulates the osmoregulation. What is the osmoregulation? It is simply the osmotic pressure. What is the osmotic pressure? That means it decides, this pressure means the pressure between the solute and the solvent. Simply, you can say that in the animal body, if the water level is very high in the body, so after that, we transfer the water by the peeing, so that the water amount should be labeled. So if the water amount is very low inside our body, so we drink water, so that the water amount should be same. So here also same function like the contractile vacuole. That means inside the amoeba, let's say suppose, the water level is very high, so it discards the water to the outside. So if the water level is very low, then it don't allow to discard the water to the outside. So this is the main function of the contractile vacuole, which is found in the protozoans, for example, the amoeba. So this is the all about the vacuoles, and this is the all about the endomembrane system, which includes endoplasmic reticulum, Golgi, lysosome, and vacuoles. Okay, so if you like the video, then please like and subscribe. It will take maximum 2 seconds. Thank you. Hello everyone. Today we will discuss about special organelle that is present in the cell. So what are the special organelle that is present in the cell? Look at the cell. In the cell there are many organelles. For example, some nucleus are present, and from the nucleus the ER is present, and then the Golgi are present, and then some ribosomes are present here. The ribosome present in the cytosol is called as the cytoplasmic ribosome, and some ribosome are also present on the rough endoplasmic reticulum, or that's why it is called as the rough endoplasmic reticulum, because in the special part of the endoplasmic reticulum here. So here some ribosomes are also present. So these ribosomes are called as the membrane bound ribosome. Or which membrane? That is the ER membrane. So this is called as the membrane bound ribosome. This is the ribosome. Some are called as the lysosome are also present. Some vacuoles are also present. Peroxisome. There are the many different organelles are present inside the cell. So what are the special organelles? So special organelles means, for example, a vacuole. A vacuole contains single membrane bound organelle, or it is called as a single membrane bound organelle. Why it is called as a single membrane bound organelle? Because let's suppose this is the vacuole and it is surrounded by only one plasma membrane or the cell membrane. This is the cell membrane. So it is surrounded by the single cell membrane. But some organelles are also present which is surrounded by the two membranes. For example, the mitochondria. The mitochondria has the two membrane That means one membrane is called as the outer membrane and another is called as the inner membrane. That means it is surrounded by the two membrane or the two plasma membrane. So that's why the mitochondria is also called as the double membrane bound organelle. Double membrane bound organelle. So besides the mitochondria, one is the mitochondria, the chloroplast. Chloroplast of the cell is also a double membrane bound organelle. That means in the chloroplast also there are two membranes. One is the outer membrane, another is the inner membrane. And one is the nucleus. So that means in the nucleus also two membranes are present. So that means these three structures, that means mitochondria, chloroplast, nucleus. So they are the different organelles or the special organelles. Why? Because these three have two membranes that are present outside the organelle. So that's why these mitochondria, chloroplast and the nucleus, they are called as the double membrane bound organelle, and all other organelles, for example, ear, Golgi, lysosome, vacuole. So they all are the single membrane bound organelle. So today we will discuss about some double membrane bound organelles. So first come to a very special structure or a special organelle that is called as the mitochondria. Now this is the mitochondria structure. So mitochondria, it is a plural term. Plural term. So that means if many mitochondria are present, then it is called as the mitochondria. There is no mitochondria. There is no term about the mitochondria because mitochondria is itself, it is a plural. So if single mitochondria is taken, then it is called as mitochondrion. So mitochondrion is a singular term. That means a single mitochondria is called as the mitochondrion, while more than one mitochondrion are present, and then it is called as the mitochondria. Okay. So now come to about some history. That means who discovered the mitochondria. So Benda, Benda, Benda in 1898, he discovered the mitochondria and they coined the term the mitochondria. This term was coined by the Benda. And mitochondria is sometimes it is also called as the parabasal bodies, or it is also called as the chondrosome, or sometimes it is also called as the plasmosome. So they are the different names of the mitochondria. That means the parabasal bodies and the chondrosome or the plasmosome. So that means they are also the mitochondria. That means they are the different terms of the mitochondria we used. But mostly we use the term mitochondria. So the mitochondria is present in all the cells except prokaryote and mature RBC. That means the prokaryotes and the mature RBC, they don't have the mitochondria structure or the mitochondria. Please note that it is very important and it is most usual need question. That means in the RBC, which structure is present or which organelles are present. So always the mature RBC is absent in the RBC. It is many times it is asked in the NEET exam that which organelles lack in the RBC, and the answer will be the mature RBC. And besides the mature RBC, prokaryotes also, they don't have this mitochondria structure. So if we go to the structure or the shape is sausage or cylindrical. The sausage it looks like this. It is a stuffed meat structure. So the mitochondria, look at the structure. It looks like exactly the sausage or the cylindrical shape of the mitochondria. So the number, if we look about the number of the mitochondria, it varies. It varies based on the different types of the physiological activities by the different types of the cell. For example, one algae is present that is called the microsterias. It is a algae and it contains only one mitochondria. That means a single mitochondria is present in the microsterias, that is the algae. For example, in the human, the liver cell contains nearly 500 mitochondria, while the kidney contains 500 to 1000 mitochondria. So the number varies according to the physiological activities by the cell. So if we look at the diameter, so this is the length, this is the diameter. So the diameter is 0.2 to 1 micrometer. Micrometer means 1 micrometer is equal to 10 to the power minus 6 meter. So the diameter is 0.2 to 1 micrometer and the length is 1 to 4.1 micrometer. This is the length and this is the diameter of the mitochondria. And the mitochondria, it is not visible easily in the compound microscope. That means the mitochondria is seen in the higher magnification, That means in the compound microscope, as it is present in the laboratory, in the compound microscope it is not seen. It is seen only in the higher magnification. And the mitochondria is also called as the semi-autonomous organelle. Semi-autonomous organelle. Why it is called as the semi-autonomous organelle? It is very, very important. Why it is called as the semi-autonomous organelle? So what is the difference between the autonomous organelle? What is the semi-autonomous? Semi means not fully. That is called as the semi. Autonomous means it doesn't depend on other cells. So that means, look at the structure of the mitochondria. It contains a special structure here like this. And this structure is called as the DNA. The DNA. That means the mitochondria also contains a DNA. That means, for example, the ER. ER doesn't have any DNA structure. For example, the Golgi. Golgi have no DNA. But the mitochondria has a DNA structure. That means if in the cell, let's say it is the nucleus. In the nucleus, a DNA is present. How many DNA? 46 or 23 pairs DNA are present. So in the, let's say this is the mitochondria. The mitochondria also contains a DNA. But this DNA is different from the DNA present in the nucleus. What is the difference? So the DNA present in the mitochondria is circular one. Circular and double stranded. Look, it means it looks like this. It is a double stranded. This is the one strand, this is the another strand, and it is the circular. But in the nucleus, the DNA is linear and double stranded. That means in the nucleus, the DNA is looks like this. That means it is the linear and double stranded. While in the mitochondria, the DNA is circular and the double stranded, and single DNA means one DNA is present in the mitochondria. So because of the presence of the DNA and along with that, some ribosomes are also present. This is the ribosome. So that means the ribosome is present, and the ribosome helps in the synthesis of the protein. That means the DNA is present, the ribosome is present. So that means this mitochondria also can synthesize its own protein. That means in the other cell, for example, this is the ER. The ER forms the protein. And this protein goes to either to the lysosome or to the peroxisome or to the vacuole. So all the proteins that are required in the different organelles, that are synthesized only in the ER. But it is the mitochondria. The mitochondria contains the DNA, and it synthesizes its own protein. That means it does not depend upon the ER for the protein part. So that's why it is called the semi-autonomous. It is not fully autonomous. Why it is fully autonomous? Because it contains only the ribosome and the DNA. But all means besides the protein, all other compounds or the other molecules, and for this it depends upon the other organelles. So that's why it is called the semi-autonomous organelles. Okay. So now look at the division. That means, let's say this is the mitochondria. This is the mitochondria. How it divides or how it doubles? So it divides by a binary fission. Binary fission. That means from one it forms two same like structure or same mitochondrial like structure. So that means always important that mitochondria divides by the binary fission. So now look at the structure of the organelle or the structure of the mitochondria. Look at the structure of the mitochondria. This is the structure of the mitochondria. And the mitochondria is the double membrane bound structure. That means one is called as the outer membrane. So this is the outer membrane. So another layer is called as it is called as the inner membrane. The outer membrane is smooth. That means it is present like straight. But the inner membrane, it forms a infolding or it moves inside it. That is called as the infolding. And this infolding structure is called as crista. Crista. Please important, the crista is the singular term, singular. If more than one crista if we say, then it is called as the cristae. And the cristae is the plural term. Okay, so what is the crista? Crista is simply the infolded structure of the inner membrane. Look at this structure. So this is the inner membrane, the infolded structure. This is called as the cristae. This is called as the cristae. This is called as the cristae. So why the cristae is important? Because inside the cristae or the cristae provides the surface area or it increases the surface area. What is the surface area? Look, this structure. Let's suppose this inner membrane, if it will goes like this. So that means this this much of distance covers. But when it infolds inside it, or when it goes to the inside, the distance is from here to here. That means nearly 3 times more than that. So that's why the infolding of the crista, it increases the surface area. So this is the outer membrane, this is the inner membrane, and the infolding part is called as the crista. So this middle part, that means between the outer membrane and the inner membrane, this space, and this space is called as the perimitochondrial space. Perimitochondrial space. So this is the outer membrane, this is the inner membrane, this is the crista, and the difference of the space between the outer and the inner membrane is called as the perimitochondrial space. And the inside it, here, this part is called as the matrix. Okay. So that means here it is the outer membrane, this is the inner membrane, this is the cristae part, and the space between the outer and the inner, this structure is called as the perimitochondrial space. And the inside it, here, this part, this inside part is called as the matrix. Okay, this is very, very important. So now come to the crista part. So the crista, or the inside part, let's suppose this is the outer membrane, this is the inner membrane. So outer membrane contains the many enzymes of the proteins, and the inner membranes also contains the many enzymes. But in the inner membrane, the important enzymes are present in the inner membrane on the surface of this inner surface. So this is called as the crista, and it contains, look at the structure, these are called as the respiratory enzymes. What is the respiratory enzyme, or what is the function of the respiratory enzymes? For example, the respiratory enzyme, it forms the energy, or it creates the energy, or it creates or generates the ATP. How the ATP is formed? By the oxidation of carbohydrate and fat. What is the oxidation of carbohydrate? What is the fat? Look at the structure. For example, a glucose is present. Okay, the glucose is converted to pyruvic acid, the pyruvate, and this process is called as the glycolysis. Always please remember that this glycolysis always occurs in the cytoplasm. Okay, so in the cytoplasm, the pyruvate is formed, and once the pyruvate is formed, it goes to the Krebs cycle. And the Krebs cycle occurs in the matrix of mitochondria. So that means one glucose is converted to 2 pyruvate, and now this pyruvate enters into the matrix part, that means the inside here, the inside part, and in the matrix the Krebs cycle occurs. So After entering this pyruvate into the mitochondria, so now in the membrane there are many respiratory enzymes are present, and these respiratory enzymes creates or forms the ATP or adenosine triphosphate. So ATP by the oxidation of this carbohydrate. That means glucose, fructose. These are the many carbohydrates. So it forms the ATP. So that's why the mitochondria is also called as the powerhouse of cell, because it creates or it generates the energy in the form of the ATP. So look at the structure. What are the respiratory enzymes? Let's suppose this structure, if I will zoom this, then it looks like this structure. That means this is the crista, and the crista different. This green structure are present, and they are called as the respiratory enzyme, which helps in the ATP formation. And along with this green structure, a yellow structure is present, and it is a very important structure. And if I zoom this, it looks like this. And this is called as the oxygen. Or this is also called as the F0, F1 particle. Or it is also called as the elementary particle. So why it is called as the oxygen or the F0, F1 particle or the elementary particle? So F0, F1, that means this top structure, this is called as the base, or it is called as the F0 part. And this round like structure, the head part is called as the F1. So that's why it is called as the F0, F1 particle, or because a stalk is present, it is also called as stalked particle. So what is the role of this oxygen? Because actually there are different types of the respiratory enzyme, and this oxygen is also a one type of respiratory enzyme, but this oxygen actually it forms the ATP. That means the respiratory enzyme forms the ATP, but out of all the respiratory enzyme, actually at the end the oxygen forms the ATP. All other respiratory enzyme helps in the generation of the ATP, but the oxygen part, it forms or it generates the ATP. So that's why the oxygen or F0, F1 particle is very important, because it creates or generates the ATP, and that's why the mitochondria is also called as the powerhouse of the cell. So because this oxygen or the oxygen, it also forms the ATP, and the ATP is formed with the, let's suppose this is a F0, F1 particle, and the oxygen is present. So it generates the ATP. So now enzyme is present, that is called as ATP synthase. So what is the role of the synthase? So the role of the ATP synthase means the ATP generates. That means this enzyme generates the ATP. So now the oxygen exactly it has the same role like the ATP synthase. So that means in the NEET exam it is also asked that which enzyme resembles the oxygen, and the answer will be the ATP synthase, because the role of the ATP synthase is to generate the ATP. The role of the oxygen is also the generation of the ATP. So that's why the role of the oxygen resembles the enzyme, the ATP synthase. Okay, so this is all about the mitochondria structure. So now come to the plastid. Come to the structure plastid. The plastid is also a double membrane bound organelle. So that means it contains two membranes. So here that means one is called as the outer membrane and another is called as the inner membrane. Okay, so that's why the plastid is also called as the double membrane bound structure. And the plastid, it can be easily visible in the compound microscope. In the compound microscope, it can be observed easily, the plastid structure. And the plastid structure, it contains specific pigment. And this pigment provides different color. So because of the providing this different color, the plastid are naming according to the color of the pigment. So the plastid are three types of the plastid are present. One is called as the chloroplast. What is the chloroplast? Another is the chromoplast. And third one is also called as the leucoplast. So what is the chloroplast? The chloro means the green. So that means it provides the green color to the obviously the plant. That means the plant contains the plastid. And the animal, they don't have the plastid structure. So green color, it provides the green color. The chromoplast means it provides the different color. Different color means the color other than the green. For example, the red, orange, different types of the colors are present. And the leucoplast is the colorless plastid. So according to on the basis of the color, the plastid are divided into three parts: chloroplast, chromoplast, leucoplast. Chloroplast provides the green color, chromoplast provides different color, and leucoplast it is a colorless plastid. So now come to the first part, that is the chloroplast. First one, that is the chloroplast. And the chloroplast found in the green plant, obviously. And in the green plant, the mesophyll cell contains the chloroplast. So the chloroplast structure, the shape, is the lens-like structure. So lens means in our eye, the lens looks like this. So here also the chloroplast also, it looks like this. Or you can be say the oval shape. For example, in some ribbon-like structure are also present. Ribbon-like structure. Ribbon-like means in the Spirogyra. In the Spirogyra, the structure is like this. That means like a ribbon-like chloroplast is present. So different types of the shape is varies. They vary. Maximum is lens of the oval shape, and some are discoid or ribbon-like structure are present. So if we look at the number, so the number actually it varies. For example, in the Chlamydomonas, the number is 1. But in the green algae, it is 20 to 40. 20 to 40 per cell. That means one cell contains 20 to 40 plastid or the chloroplast in the green algae. In the plant, there are many, more than 20 or 40 chloroplast in the plant. So the plastid is also called as the semi-autonomous organelle. Why it is called as a semi-autonomous organelle? Because it contains a DNA. Look at this structure. So this DNA is circular and the double stranded. So that means in the nucleus, the chromosome is the linear double stranded, while the DNA present in the chloroplast is double stranded but circular and the single. That means one circular double stranded DNA is present. Like in the mitochondria also contains single circular double stranded DNA structure is present. And according to that, and beside that, this structure like that are present. And this structure is called as the ribosome. And this ribosome is called as cytoplasmic ribosome. So what is the cytoplasmic ribosome? Look at the structure. Let's say this is the nucleus, and from the nucleus the ER is present. So in the ER or the rough endoplasmic reticulum, some ribosomes are also present, and some ribosomes are also present in the cytoplasm. So the ribosomes that are present on the membrane of the ER, so they are called as the membrane bound ribosome. And the ribosome present in the cytosol is called as the cytoplasmic ribosome. So that means because the ER is not present in the chloroplast, so that's why only the cytoplasmic ribosome is present in the chloroplast. So because the presence of the DNA in the ribosome, so it can synthesize its own protein. So that means it does not depend upon the other organelles for the protein part. So that's why it is called as a semi-autonomous organelle. But beside the protein, other molecules, and for other molecules, this cell depends upon the other organelles. So only for the protein, it is independent, and that's why it is called the semi-autonomous. Semi means not fully. So that is called the semi-autonomous organelle. Okay. So now look at the structure of the. Come to the structure of chloroplast. So the chloroplast is the double membrane structure. That means the outer is called as the outer membrane, and the inner part is called as the inner membrane, because of the presence of the two membranes. That means outer and the inner. That means the two membranes are present. That's why the plastid are also called as the double membrane bound structure. And now look at the structure. This structure, that means this is a space. The space between outer and the inner membrane is called as the intermembrane space. Okay. So now come to this structure. This inside it is called as the matrix. That means inside the inner membrane, all the structure is called as the matrix. So in the matrix, there are the different types of the structure is present. One is called as the grana. One is called as the thylakoid. One is called as the stroma. So what are the different types of this structure? So what is the grana? Look at this structure. This whole structure is called as the grana. And again, grana is the plural term. The singular term is granum. Okay. So this is the grana. So what is the thylakoid? Look at the structure of the grana. So it is, there are many stacked particles present, and each one is called as the thylakoid. So that means the different or many thylakoids, they combine together to form the grana. So this is the thylakoid. What is the stroma? Stroma is the liquid part. Inside it, the grey part is called as the stroma. Okay. So now clear what is the grana, what is the thylakoid, what is the stroma. So grana means the granum. This is the one granum. This is the one granum. All total are called as the grana. That is the plural term. So in the grana, it is formed by the many thylakoid structure, and the liquid part is called as the stroma. So now look at the structure. This grana is joined by a structure. Here look like this structure. Here this structure. Here this structure. So the linking structure which links different grana are called as stroma lamellae. Stroma lamellae. Okay. So this is all about the different types of the structure. So now come to the grana part. What is the function of the grana? What is the function of grana? So when from the sunlight or the sun, the light comes to the, let's suppose, leaf. There are many in the leaf, there are many cells, and inside the cell there are many organelles. So these organelles, one is called as the chloroplast, and the chloroplast, the granum, actually this granum or the grana, it actually absorbs the sunlight. Only the grana of the cell of the plant absorbs the sunlight, and it performs the, most importantly, photosynthesis. The photosynthesis occurs actually in the chloroplast, and in the chloroplast actually the grana part, it forms the photosynthesis. So because of this, the grana is also called as the site of light reaction. Why it is called as the site of light reaction? That means in the presence of the light, some reactions are obviously present, and that is called as the photosynthesis. So that's why, most importantly, the grana is called as the site of the light reaction. That means it performs the photosynthesis. So now come to the stroma. What is the function of the stroma, the watery part or the liquid part? So this is the stroma, or the stroma is the watery part. Obviously, it contains some enzyme, and that enzymes forms the carbohydrate and protein. Okay, how the protein is formed? Because on the help of this double-stranded circular DNA along with ribosome, and this formation of the carbohydrate and the protein, it forms in the light, in the dark. That means in the night. So that's why the stroma is also called as the site of dark reaction. site of dark reaction. So site of the light reaction is the grana part, and the stroma or the liquid part is called as the site of dark reaction. So this is the all about the chloroplast. So now come to again another structure or another plastid that is called as second part, that is the chromoplast. What is the chromoplast? That means it provides different types of the color. For example, there are many types of the chloroplast. For example, the carotene. Okay. So carotene, other is the xanthophyll. So the carotene provides the orange color. And the carotene is present in the carrot. So that's why the name is the carotene. And it comes from the carrot. Actually, the color of the carrot is also the orange. And this orange color of the carrot is due to the carotene, because the carotene is the chromoplast which provides the color. So like that in the xanthophyll it is the yellow color. So it is mainly present in the young leaves, and besides the young leaf, many vegetables, they also contain the xanthophyll like plasmid. So this is the chromoplast. And the third one is called as the leucoplast. So leucoplast means it is a colorless. That means it does not provide any color to the plant. So what is the function of the leucoplast? It helps in the storage. So it stores many molecules. For example, there are many types of the leucoplast, and on the basis of the storage item, the leucoplast are divided into many parts. One is called as the amyloplast. So the amyloplast, it stores the starch, which is the carbohydrate. Okay, so the leucoplast, which stores the carbohydrate or the starch, is called as the amyloplast. Another is called as the elioplast. The elioplast, it stores the oil and fat. The elioplast, it stores the oil and the fat. And another is the eleuroplast. And it stores the protein. So different types of the leucoplast are present, which stores different types of the molecule. Some contain the starch, it is called amyloplast. Some stores the oil and the fat, and it is called the elioplast. And some contains the protein, and it is called the aleuroplast. So different types of the leucoplast are present, and this is all about the plastid. That means the chloroplast, chromoplast, and the leucoplast. So if you like the video, please like and subscribe my video. Thank you. Hello everyone. Today we will know about the ribosome. So what is the ribosome? So the ribosome is simply, it is an organelle. So like the ER, like the nucleus, like the lysosome. So the ribosome is also an organelle. And the ribosome is membraneless organelle. Membraneless organelle. So what is the membraneless organelle? So that means the ribosome doesn't have any membrane. So So like for example the endoplasmic reticulum, for example the Golgi, so the nucleus, like the mitochondria, so they all have a membrane, but the ribosome, it doesn't have any membrane, so that's why the ribosome is also called as a membraneless organelle. So first, the discovery of when the ribosome was discovered, the answer is 1953. But in the animal, the George Pelled discovered the ribosome in the animal cell in the 1953, while in the same year in the plant, Robinson and Brown, so they discovered the ribosome in the plant in same year, that is the 1953. So that means the ribosome was discovered in the 1953, but in the animal, the George Pelled discovered the ribosome, but in the plant, the Robinson and the Brown, they discovered the ribosome in the plant cell. So now the ribosome is also called as the protein factory. Protein factory. Why it is called as the protein factory? Because the ribosome is also, it helps in the formation of the protein. So that's why the ribosome is also called as the protein factory, or without ribosome the protein cannot be formed. So that's why the ribosome is also called as the protein factory, or it is also called as the engine of the cell. Engine of the cell. Okay, so why it is called the engine of the cell? The same, like the for any instrument, engine is required, and here the engine is the ribosome, because it forms the protein which is most necessary for a cell. So that's why the ribosome is also called as the protein factory or the engine of the cell. So now look, what is the component of the ribosome, or how the ribosome is formed. The ribosome is formed by rRNA plus protein. So that means in the ribosome, RNA is present and the protein is present in the 2:1 ratio. That means the rRNA is present double in comparison to the protein. So the rRNA and the protein is formed, or the protein is present in the ribosome, and because the rRNA is called as the nucleo, or it is present in the nucleus, so it is the nucleo, it is the protein, and this is the protein. So that's why the ribosome is called as the nucleoprotein, because it has the protein and it has the RNA, or the nucleus part. So that's why the ribosome is also called as the nucleoprotein, and the ratio is the RNA is present double in comparison to the protein. So now look at the structure. Structure of the ribosome. So the ribosome is simply a spherical shape. The spherical shape with a 150 to 250 angstrom, that is the total volume is the 150 to 250 angstrom. So angstrom is 10 to the power minus 9 meter. So that is the angstrom. So 150 to 250 angstrom is the main area of the ribosome. And if we zoom this ribosome, and it looks like this. So that means it has one larger subunit, and that is called as the large subunit, and it has a smaller subunit, and that is called as the small subunit. Okay. So now the large subunit and the small subunit, they are bound together with the help of the magnesium. This is very important. With the help of the magnesium, the large subunit and the small subunit, they are attached to each other. So the ribosome subunit are the unequal. That means the subunits are not in the same size. That means one is the larger one, another is the smaller one. And the larger one is called as the large subunit, and the smaller one is called as the small subunit. So now look at the types of ribosome. So the ribosome can be bound, can be free. The bound is also sometimes called as the membrane bound ribosome, and the free ribosome is also called as the cytosolic ribosome. So what is the membrane bound ribosome? What is the cytosolic ribosome? Look at the structure. Let's suppose this is cell and this is the nucleus, and from the nucleus the ER is projected from the nucleus, and after that the Golgi body is present. So now some ribosome are present here on the membrane or on the membrane of ER, and that is called as the membrane bound ribosome. And that structure of the ribosome, so structure of the ER is also called as the rough endoplasmic reticulum. So ER has two types. One is called as the rough endoplasmic reticulum, one is called as the smooth endoplasmic reticulum. So the rough endoplasmic reticulum, why it is called the rough? Because of the presence of the ribosome on the membrane of ER, and that's why this structure is called as the rough endoplasmic reticulum. While this portion of the ER, it doesn't have any ribosome in its membrane, so that's why it is called the smooth endoplasmic reticulum. So that is the bound of the membrane bound ribosome. But what is the free or the cytosolic? Because some ribosome are also present on the cytosol, and that is called the free. That means it is not membrane bound. So that's why it is called the free or cytosolic. Why it is called as cytosolic? Because it is present in the cytosol. So this is the main two types of the ribosome. One is the membrane or the membrane bound ribosome that is present on the membrane of the ER, and the another is the free or the cytosolic ribosome. So now come to the main one is the membrane bound, another is the cytosolic, and another types of the ribosome. One type is called as the 70S ribosome, and another is called as the 80S ribosome. So what is the S? So S is the unit of the ribosome, and it is also called as the Swedberg unit. So Swedberg, he was a scientist, and he discovered this ribosome subunit. So on the name of this scientist, it is called as the S, so the Swedberg subunit. But sometimes the S is also called as the sedimentation coefficient. Sedimentation coefficient. So S stands for the Swedberg unit, or you can call it as the sedimentation coefficient, which is the unit of the ribosome. So now the different types of the ribosome, one is the 70S, one is the 80S. So what is the difference between the 70S and the 80S? So normally the 70S, it has also the structure like this. That means it has also a large subunit, another is the small subunit. But the large subunit is 50S, while the small subunit, it is the 30S. That means the 70S ribosome, it has a large subunit, it has a small subunit. The large subunit is a 50S and the small subunit is a 30S. And if in the 50S, then 28S ribosome is, or it is the main component of the 50S. So that means in the 50S of the large subunit, a 28S subunit is main component of the 50S, while in the 30S, 18S is the main component of the 30s. It is very important. Please remember that. And don't add, that means 50 plus 30, it is the 80. It is not of the 80, it is the 70s. It is according to the molecular weight. So please remember that in the 70s, the large subunit is of 50s and the small subunit it is of 30s. And in the 50s, the main component is the 28s, and in the small subunit, the main component is the 18s. So now, in the 80s, it is also a large subunit and a small subunit. So the large subunit, it is the 60s one, and the small subunit, it is the 40s one. And if we find what is the main component in the 60s of the large subunit, and that is the again the 28s plus 5s. Or in the 40s, the main component is the 18s. Okay. So now, what is the 70s, what is the 80s, you know, but where the 70s and where the 80s ribosomes are present. So the 70s ribosomes are present in the prokaryote along with some organelle of our body, for example, the chloroplast, mitochondria. So they are the 70s type. While in the 80s, it is found mainly in the eukaryote. So in the eukaryote, for example, in our case, human being is also eukaryote. So in the eukaryote, the 80s ribosomes are present, or in the higher animals, the 80s ribosomes are present, but in the 70s is present in the prokaryote, like the bacteria, archibacteria and the eubacteria, they are the 70s. But in the eukaryote, we have also the 70s type, but that is present in the chloroplast and that is present in the mitochondria. But the 80s is present in the mainly in the membrane of the ribosome, and that is called the rough endoplasmic reticulum. And that rough endoplasmic or that ribosome that is present on the membrane of the ear is the 80s type. While, for example, look at this is the cell, this is the nucleus, this is the ribosome. Let's suppose this is the Golgi body and this is the rough endoplasmic reticulum. So now the ribosome is present and this one is called as the 80s, and this is the eukaryotic cell. And in the cell also the mitochondria is present, and inside the mitochondria some ribosomes are present, and that is called as the 70s. For example, in the chloroplast it is present in the plant cell. So in the plant cell also the chloroplast, and inside the chloroplast the ribosome is present, and they are also the 70s. So in the eukaryote the 80s are present on the membrane of the ribosome and the chloroplast in the mitochondria. While in the prokaryote, for example in the prokaryote, this is only a nucleus is present, not nucleus is present, and along with that a ribosome is simply present, and this ribosome is called as the 70s, and this is the prokaryote cell. So in the prokaryote cell 70s is present, and the eukaryote 80s is present on the membrane of the ear, and that forms the protein, while the 70s is also formed in the is present in the mitochondria and the chloroplast, and they also helps in the protein synthesis in the mitochondria and in the chloroplast also. So this is the main types of the ribosome. So now if we go to the biosynthesis. So what is the biosynthesis or a biogenesis? What is the biogenesis? Biogenesis means where the ribosome is formed. So mainly the ribosome is formed in the prokaryote. In the prokaryote. eukaryote, it is the ribosome is found obviously in the cytosolic. This is very very important, and this is a neat question. While in the eukaryote, the ribosome is present in the nucleus. The ribosome is found in the nucleus. So the biogenesis of where the ribosome is found in the eukaryotes, inside the nucleus the ribosome is found, while in the prokaryote, in the cytosol, the ribosomes are found. So now a term is called the polyribosome. Polyribosome, or it is also called as the polysome. Simply it is called as the polysome or the polyribosome. So what is the meaning of this? That means in the protein synthesis or during the protein synthesis, so the many ribosomes are bound to the, this is the mRNA, and the protein is formed from the mRNA, and the mRNA is bound to the protein, bound to the ribosome. So here the many ribosomes are attached to the mRNA, and that is called the poly, means many, and the ribosome, that means many ribosomes are formed or are organized during the protein synthesis. So that is the condition where it is called as the polyribosome or the polysome. And then the eukaryote, this is the large subunit, this is the small subunit. The large subunit and the small subunit, they are bound together, and the one main enzyme is important, that is called as the peptidyl transferase. So that means a peptidyl transferase, it is main important for the bound or for binding of the large subunit with the small subunit, and it is mainly bound by the magnesium, or the magnesium or the peptidyl transferase, it needs the magnesium for the bound of the large subunit and the small subunit. And the main function of the ribosome is to form or to produce the protein. That is the main function of the protein. So this is all about the ribosome, and if you like the video, and please like and subscribe my video. Thank you. Hello everyone. Today we will know about the cytoskeleton structure. So first of all, what is cytoskeleton? Cyto means the cytoplasm, and the skeleton means, of course, the skeleton. That means inside of the cytoplasm, a skeleton-like structure is present, and that is called as the cytoskeleton. So first of all, just imagine an invertebrate animal. So an invertebrate animal. So in the invertebrate animal, that means it doesn't have any vertebral column. So without a vertebral column, so that means the animals should be just like a balloon which is filled with the water. So that means it may, or it should shrink like this, or it shrink like this, because it doesn't have any vertebral column. But it is present like this, or they live like this. So why? Because of the presence of the cytoskeleton. So that means in the invertebrate as well as in the vertebrate, the cytoskeleton, it also works like the vertebral column. Vertebral column. How it works like the vertebral column? Because it provides the support to the cell, or to the body, or to the organisms. So that is the main function of the cytoskeleton, that means to provide the support. And besides providing the support, it also helps in the motility inside the cell, and along with that it also helps in the maintenance in the cell shape. That means the cell shape can be determined or can be possible due to the presence of the cytoskeleton. So now What are the different types of the cytoskeleton that is present inside our organism? So there are the three types of the cytoskeleton are present. One is called as the microtubule. The second one is called as the microfilament, and the third one is called as the intermediate filament. So these three are the different types of the cytoskeleton that is present inside our body. Of course, these three have different type of the role inside our body. So the main function of the cytoskeleton, or the main function of these three cytoskeleton, are the support to the cell or to the body. So first look at the microtubule. So what is the function of the microtubule? So the main function is to help in formation of mitotic spindle fiber. Mitotic spindle fiber. So what is the mitotic spindle fiber? So that means, for example, in the cell or during the cell division, so the spindle fiber, let's suppose this is the chromosome in the metaphase state, and in the metaphase state a microtubule spindle formation occurred, and this spindle formation and this spindle are made up of the microtubule. So it also helps in the formation of the also helps in the cell division by forming the mitotic spindle fiber formation. The second one is it also helps in the distribution of pigment. Distribution of pigment. So for example, in the plant, suppose the chloroplast, or in the animal, suppose the melanocyte. So they all are the pigment structure that is present in the different organisms. So these pigments are distributed by this microtubule. The third function of the microtubule is the cell differentiation. Cell differentiation. So what is the cell differentiation? For example, let's suppose this is an immature cell. So now it is converted to the, for example, the neuron cell. For example, it is converted to the sperm cell. It may converted into the excretory cell, or like the phloem cell. So that means different cell differentiation are occurred, and it is due to the microtubule. And the fourth function is the cell movement. Cell movement. For example, suppose during the gastrulation, one cell moves to another, and this cell movement is occurred or is provided by the microtubule. And the main function, of course, it also helps in the cell support. That means it supports, it provides the support to the cell. So these are the main function of the microtubule. So now we should know about the microtubule extensively. So the microtubule, it is formed by the tubulin plus GTP, or the guanine triphosphate, plus Mg plus 2, or the magnesium. So that means the main component of the microtubule is the tubulin. So please think like the tubulin structure is like that. So that means the tubulin structure, it is a dimer structure. So what is the dimer? That means it has two components. One component, another is this is the another component. So this is the dimer. So now this is the one tubulin. So now one tubulin, or to the one tubulin, another tubulin is joined. And this joint is accompanied by the GTP with the help of the, or with catalyzation of the magnesium. That means With the help of that, it needs the GTP, and it also needs for the magnesium for the activation of the GTP. So with the help of the GTP and the magnesium, one tubulin joins to another tubulin. So now to this another tubulin are joined. Now another tubulin are joined. So like that a strand is formed. And now this strand is called as the protofilament. Protofilament. So now to this protofilament, this is the one protofilament, and to this one protofilament another protofilament is joined. Okay, and to this another protofilament are joined. So like this 13 protofilaments are joined together. So like this is the structure of the microtubule. That means here this is the one protofilament, this is the second protofilament, this is the third protofilament. So like that total 13 protofilaments are joined together and it forms a tube-like structure like this. Okay, so this is called as the microtubule structure. So in the 3D structure it looks like the tube like this. This is the 3D structure of the microtubule. So now if you take the cross section, for example this structure, if we take the cross section it looks like this. So that means 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilaments. So that means this is the 1, 2, that means 13 protofilaments are present. So in the cross section it looks like this. So in the book you look like this, like structure, and this is the cross section of the protofilament, or this is the cross section of the microtubule like structure. So now come to where the microtubule are found. So the microtubules are found in the cilia, flagella, as well as in the centrioles. So first come to the cilia and the flagella. Cilia, another is the flagella. So what is the cilia and what is the flagella? They are the locomotory organ. What is the locomotory organ? That means it helps in the locomotion of the organism. For example, the cilia in the paramecium. In the paramecium, the cilia, this like structure is present and they are called as the cilia. For example, the euglena. In the euglena, a tail-like structure is present and that is called as the flagella. So that means the cilia, it is the singular term is called as the cilium. That means one cilia is called as the cilium, while one flagella is called as the flagellum. Okay. So now the number of the cilia are always high, but the number of the flagella maximum 1 or the 2, or mostly the one flagella is present, or the single flagella is present, or the single flagellum is present, or maximum 2 flagella are present. For example, in the euglena, the flagella is present. In the paramecium, that means it looks like the slipper. So that's why the paramecium is also called as the slipper-like animalcule. So in the paramecium, the cilium is present, and in the euglena, the flagella is present. So now, in the cilia, or the animals which are the cilia, so that means all the cilia must have moved at a time so that it can move to the forward or to the backward. But in the flagella, because of the presence of single or maximum to 2, that means it is not coordinately mannered. That means it can move independently, while the cilia must have coordinately move to each other so that organism can move to forward either way or backward. So now the cilia it runs like a running manner, or it moves like a running manner. Running manner means all should have, or all runs like this, but in the flagella it presents, or it moves like wave-like manner. Wave-like manner. So this is the main difference between the cilia and the flagella. So now look at the structure of the cilia and the flagella, or the microtubule which is present in the cilia and the flagella. And but before going to that, for example, this is the paramecium. Okay. So the cilia structure, it looks like this. So that means this is the whole cilia structure. So now some part of the cilia is protruded in the cytoplasm, and some are present in the outside. So the outside part of the cilia is called as the axoneme. This is very important, axoneme. While the part which is present inside the cytoplasm or inside the body, that is called as the basal body. Again, it is the same for the flagella. That means flagella also, it has the same structure. That means the outside flagella structure is called as the axoneme, while the structure which is present inside the body or which is present in the cytoplasm is called as the basal body. So this is the main difference between the cilia and the flagella structure. That means the cilia and the flagella structure, they have two different structure. One is called the axoneme, another is the basal body. And the structure of the axoneme and the basal body is that obviously the microtubules are present inside the axoneme or inside also in the basal body, but the structure are very different. So now look at the structure. So now first we should look about the axoneme structure. So now I am telling about the axoneme structure. That means the structure which is present outside the body is called as the axoneme structure. So now in the axoneme structure, the structure looks like this. So that means here they are called as the peripheral ring. So what are these structure? For example, as we have discussed earlier, this structure. So here, for example, two ring-like structure is present. So that means one structure is like that. That means 13 protofilaments are present. And to that, again, one, for example, this is the one tube, and to this, another tube are joined. Same like structure. So that means if we take the cross section, it looks like this. That means one, another is another structure. So this is the main structure of like this. So this is called the peripheral ring. So in the axoneme, the outside ring are called the peripheral ring, and they have 9 peripheral rings. This is the first, this is the second, this is the third, fourth, fifth, sixth, seventh, eighth, and the ninth. So that means 9 protofilament like structure, the 9 microtubule structure is present in the peripheral, and in the central region, this is the central region, this is the central region, or it is called as the central ring. And in the central region, 2 structures are present. One, another is a 2. But here they are present like in the diploid structure or the doublet structure. This is the doublet structure. So the axoneme structure, as we, or if we look the structure, the axoneme structure, it is designated as 9 plus 2 arrangement. What is the 9 plus 2 arrangement? So 9 means in the peripheral structure, 9 structure or the 9 microfilament structure is present, and which are the doublet. But here the two, that means the single structure, and that is present in the twice. This is the one single structure, this is the another single structure. That means two single, that means it is the two singlet. So that means 9 doublet and 2 singlet. And the 9 doublet are present in the periphery region, and the 2 singlet structure are present in the central region, and the all overly it is called as the 9 plus 2 arrangement, and it looks like this. So now this is the central region, and in the central region the central link is joined to the peripheral link by this structure, and this structure is called as the radial spoke. So that means the radial spoke, it joins to the peripheral link to the central link, while the peripheral links, they are again joined by the connectors. These are by connectors. So these connectors are called as the nexin. So these are the connectors, or they connect the peripheral link structure. So now to this peripheral link structure, a structure like this structure is present. Here the 3 dot, if we series, so this 3 dot structure, this is main important structure, that is called as the dynein arm. So this dynein arm, it helps in the, or it releases the energy for the movement of the organism. So that means for movement some energy is required, and this energy is released by this dynein structure. So this is the all about the axoneme structure. So now come to another structure, that is called as the basal body. So that means, for example, this is the paramecium, this is the cilia structure, and this inside the body or in the cytoplasm structure, the centrioles are present. They are called as the centrioles. So now in the basal body, the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So what is the ciliogenesis? So that means inside of the body, or the inside, not our body, that means inside the, for example, the paramecium or the euglena. So this structure, so here the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So during the ciliogenesis, the centrioles are called as the basal body. So this is the again another structure of the cilia and the flagella, which is present inside the body or inside the cytoplasm. So now the structure of the basal body is different. Look at the structure, and this is same as present in the centrioles. So first, before going to the basal body structure, first we should know something about the centrioles. So what is the centriole? So for example, in the cell or during the cell division, for example in the metaphase stage, they are the chromosome which is present in the metaphasic plate. So now the spindle fibers are formed here. So this structure is called as the centrosome. So now what is the centriole, what is the centrosome? So now if we see the structure, the centrosome, here it looks like this. So this is the centrosome. So in the centrosome, two centrioles are present, and they are present perpendicularly to each other. So they are called as the centriole. So in one centrosome, two centrioles are present and they are present perpendicularly to each other here. And besides this centriole, some amorphous structure is present and they are called as the PCM. PCM means pericentriolar matrix. Just imagine, or just like the cytoplasm. So that means inside the centrosome, two centrioles are present and they are present perpendicularly to each other, and now the PCM or the pericentriolar matrix are present. So now, if we look at the centriole structure, let's suppose this is one centriole. So in the centriole, the structure looks like this. Just imagine. So that means in the axoneme, two structures are present like this here. So that means, for example, this structure, that means one tube and it is joined to the another tube. If we take cross section, it looks like this. So here, three tubes are joined to each other. 1, 2, 3. So just like that, 9 peripheral structure, 9 peripheral structures are present. So in the axoneme, this is the axoneme structure. So now in the basal body, it looks like this. That means this is the axoneme structure, or this is the basal body, which is similar to the centriole. Okay. So in the basal body of the centriole, the same structure like present, but what is the difference in the axoneme? So the peripheral link are called doublet. So they are the doublet. That means they are present two ring structure, but they are the triplet. So that means they are present or the peripheral link same. It is called as the peripheral ring structure, and this is called as the middle part. They are called as the central ring structure, or the central link or central structure, which is also called as the hub. So in the peripheral ring structure, the triplet microtubules are formed, while in the axoneme structure, the doublet microtubule structure are found. And another difference is that inside the central region, there is no microtubule structure. But in the axoneme structure, they have two singlet structures present. So that's why the axoneme structure is called as the 9+2 arrangement, and the basal body or the centriole is called as the 9+0 arrangement. And here the difference is the 9 are the doublet, and here the 9 are the triplet. This is the main difference between the axoneme structure and the basal body structure. So now look at the structure here. So it looks like the bullock cart wheel structure. That means let's suppose this is the bullock cart. This is the one wheel, and inside it another wheel-like structure. So here the centriole is also looks like same as bullock cart wheel structure. So that's why the centriole is also called as the bullock cart wheel. This is the very, very important, and In the previous exam, this question was arised that which structure is also called as the bullock cart wheel structure, and the answer will be the centriole. So this is the all about the structure or the difference between the axoneme and the basal body. So the basal body, it is same as the centriole as in the basal body structure. Here the centriole forms the cilia and the flagella, and the process is called as the ciliogenesis. So now see, this is the centrosome. This is the another centrosome. Here in the centrosome, two centrioles are present perpendicularly. Here also two centrioles are present perpendicularly. So now actually first in the cell one centrosome are present. But for example in the cell cycle, this is the M phase, this is the G1 phase, this is the S phase, this is the G2 phase. M phase means division phase, that means mitosis or meiosis. G1 means this is the gap 1. This is the S phase, that means here the DNA is synthesized, or the DNA becomes double. So now this is the G2, that means gap 2, that means the gap between mitosis or the meiosis and to the synthesis phase. So during the gap 2 phase, so now the centrosome becomes the double. So the centrosome actually in the cell, the centrosome are present 1. The number of the centrosome are 1, but during the gap 2 phase, the centrosome becomes double, just like the binary fission. So in the gap 2 phase, it becomes the double, and now it comes to one end and another is come to another end, and now the mitosis and the meiosis occurs. So now the spindle fibers are formed. So this is the all about the centrioles or the centrosomes. So now come to what is the function of the cytoskeleton, or what is the function of the microtubule. As I have already discussed, but just as a summary I am telling you that one is the spindle fiber formation. The second one is the formation of axoneme and basal body of cilia and flagella. Third one is the it also helps in the cell differentiation. So that means which cell should convert to which cell, and it is determined by the microtubule. Another is the it also distributes to the cell pigment. That means, for example, the chloroplast or the melanocyte. And another is that it also helps in the cell movement. That means during the gastrulation, one cell goes to another, or one cell again come to here. So this is the cell movement, and this cell movement is occurred by the microtubule, and most importantly, it provides the support to the cell or to the organisms. So this is all about the structure of the microtubule. So if we like the video, then please like and subscribe my video. Thank you. Hello everyone. Today we will know about the cytoskeleton structure. So first of all, what is cytoskeleton? Cyto means the cytoplasm, and the skeleton means, of course, the skeleton. That means inside of the cytoplasm, a skeleton-like structure is present, and that is called as the cytoskeleton. So first of all, just imagine an invertebrate animal. So an invertebrate animal. So in the invertebrate animal, that means it doesn't have any vertebral column. So without the vertebral column, so that means the animals should be just like a balloon which is filled with the water. So that means it may, or it should shrink like this, or it shrink like this, because it doesn't have any vertebral column. But it is present like this, or they live like this. So why? Because of the presence of the cytoskeleton. So that means in the invertebrate as well as in the vertebrate, the cytoskeleton, it also works like the vertebral column. Vertebral column. How it works like the vertebral column? Because it provides the support to the cell or to the body or to the organisms. So that is the main function of the cytoskeleton. That means to provide the support. And besides providing the support, it also helps in the motility inside the cell, and along with that it also helps in the maintenance in the cell shape. That means the cell shape can be determined or can be possible due to the presence of the cytoskeleton. So now, what are the different types of the cytoskeleton that is present inside our organism? So there are the three types of the cytoskeleton are present. One is called as the microtubule. The second one is called as the microfilament, and the third one is called as the intermediate filament. So these three are the different types of the cytoskeleton that is present inside our body. Of course, these three have different type of the role inside our body. So the main function of the cytoskeleton, or the main function of these three cytoskeleton, are the support to the cell or to the body. So first look at the microtubule. So what is the function of the microtubule? So the main function is to help in formation of mitotic spindle fiber. Mitotic spindle fiber. So what is the mitotic spindle fiber? So that means, for example, in the cell or during the cell division, so the spindle fiber, let's suppose this is the chromosome in the metaphase state, and in the metaphase state a microtubule spindle formation occurred, and this spindle formation and this spindle are made up of the microtubule. So it also helps in the formation of the also helps in the cell division by forming the mitotic spindle fiber formation. The second one is it also helps in the distribution of pigment. Distribution of pigment. So for example, in the plant, suppose the chloroplast, or in the animal, suppose the melanocyte. So they all are the pigment structure that is present in the different organisms. So these pigments are distributed by this microtubule. The third function of the microtubule is the cell differentiation. Cell differentiation. So what is the cell differentiation? For example, let's suppose this is an immature cell. So now it is converted to the, for example, the neuron cell. For example, it is converted to the sperm cell. It may converted into the excretory cell or like the plain cell. So that means different cell differentiation are occurred, and it is due to the microtubule. And the fourth function is the cell movement. Cell movement. For example, suppose during the gastrulation, one cell moves to another, and this cell movement is occurred or is provided by the microtubule. And the main function, of course, it also helps in the cell support. That means it supports, it provides the support to the cell. So these are the main function of the microtubule. So now we should know about the microtubule extensively. So the microtubule, it is formed by the tubulin plus GTP or the guanine triphosphate plus Mg plus 2 or the magnesium. So that means the main component of the microtubule is the tubulin. So please think like the tubulin structure is like that. So that means the tubulin structure, it is a dimer structure. So what is the dimer? That means it has two components. One component, another is this is the another component. So this is the dimer. So now this is the one tubulin. So now one tubulin, or to the one tubulin, another tubulin is joined. And this joint is accompanied by the GTP with the help of the, or with catalyzation of the magnesium. That means with the help of it needs the GTP, and it also needs for the magnesium for the activation of the GTP. So with the help of the GTP and the magnesium, one tubulin joins to another tubulin. So now to this another tubulin are joined. Now another tubulin are joined. So like that a strand is formed. And now this strand is called as the protofilament. Protofilament. So now to this protofilament, this is the one protofilament, and to this one protofilament another protofilament is joined. Okay, and to this another protofilament are joined. So like this 13 protofilaments are joined together. So like this is the structure of the microtubule. That means here this is the one protofilament, this is the second protofilament, this is the third protofilament. So like that, total 13 protofilaments are joined together and it forms a tube-like structure like this. Okay, so this is called as the microtubule structure. So in the 3D structure, it looks like the tube, like this. This is the 3D structure of the microtubule. So now, if you take the cross section, for example, this structure, if we take the cross section, it looks like this. So that means 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 protofilaments. So that means this is the 1, 2, that means 13 protofilaments are present. So in the cross section, it looks like this. So in the book, you look like this, like structure, and this is the cross section of the protofilament, or this is the cross section of the microtubule like structure. So now come to where the microtubule are found. So the microtubules are found in the cilia, flagella, as well as in the centrioles. So first come to the cilia and the flagella. Cilia, another is the flagella. So what is the cilia and what is the flagella? They are the locomotory organ. What is the locomotory organ? That means it helps in the locomotion of the organism. For example, the cilia in the paramecium. In the paramecium, the cilia, this like structure is present and they are called as the cilia. For example, the euglena. In the euglena, a tail-like structure is present and that is called as the flagella. So that means the cilia, it is the singular term is called as the cilium. That means one cilia is called as the cilium, while one flagella is called as the flagellum. Okay. So now the number of the cilia are always high, but the number of the flagella maximum one or the two, or mostly the one flagella is present, or the single flagella is present, or single flagellum is present, or maximum two flagella are present. For example, in the euglena, the flagella is present. In the paramecium, that means it looks like the slipper. So that's why the paramecium is also called as the slipper-like animalcule. So in the paramecium, the cilium is present, and in the euglena, the flagella is present. So now, in the cilia, or the animals which are the cilia, so that means all the cilia must have moved at a time so that it can move to the forward or to the backward. But in the flagella, because of the presence of single or maximum to two, that means it is not coordinately manner. That means it can move independently, while the cilia must have coordinately move to each other so that organism can move to forward, either way, or backward. So now the cilia, it runs like a running manner, or it moves like a running manner. Running manner means all should have, or all runs like this, but in the flagella it presents, or it moves like wave-like manner. So this is the main difference between the cilia and the flagella. So now look at the structure of the cilia and the flagella, or the microtubule which is present in the cilia and the flagella. And but before going to that, for example, this is the paramecium. Okay. So the cilia structure, it looks like this. So that means this is the whole cilia structure. So now some part of the cilia is protruded in the cytoplasm and some are present in the outside. So the outside part of the cilia is called as the axoneme. This is very important, axoneme. While the part which is present inside the cytoplasm or inside the body, that is called as the basal body. Again, it is the same for the flagella. That means flagella also, it has the same structure. That means the outside flagella structure is called as the axoneme, while the structure which is present inside the body or which is present in the cytoplasm is called as the basal body. So this is the main difference between the cilia and the flagella structure. That means the cilia and the flagella structure, they are two different structure. One is called the axoneme, another is the basal body. And the structure of the axoneme and the basal body is that obviously the microtubules are present inside the axoneme or inside also in the basal body, but the structure are very different. So now look at the structure. So now first we should look about the axoneme structure. So now I am telling about the axoneme structure. That means the structure which is present outside the body is called as the axoneme structure. So now in the axoneme structure, the structure looks like this. So that means here they are called as the peripheral ring. So what are these structure? For example, as we have discussed earlier, this structure So here, for example, two ring-like structure is present. So that means one structure is like that. That means 13 protofilaments are present. And to that, again, one, for example, this is the one tube, and to this, another tube are joined. Same like structure. So that means if we take the cross section, it looks like this. That means one, another structure. So this is the main structure of like this. So this is called the peripheral ring. So in the axonym, the outside ring are called the peripheral ring, and they have 9 peripheral rings. This is the first, this is the second, this is the third, fourth, fifth, sixth, seventh, eighth, and the ninth. So that means 9 protofilament like structure, the 9 microtube structure is present in the peripheral, and in the central region, this is the central region, this is the central region, or it is called as the central ring. And in the central region, 2 structures are present. One, another is a 2. But here they are present like in the diploid structure or the doublet structure. This is the doublet structure. So the axonym structure, as we, or if we look the structure, the axonym structure, it is designated as 9 plus 2 arrangement. What is the 9 plus 2 arrangement? So 9 means in the peripheral structure, 9 structure or the 9 microfilament structure is present, and which are the doublet. But here the 2, that means the single structure, and that is present in the twice. This is the one single structure, this is the another single structure. That means 2 single, that means it is the 2 singlet. So that means 9 doublet and 2 singlet. And the 9 doublet are present in the periphery region, and the 2 singlet structure are present in the central region, and the all overly it is called as the 9 plus 2 arrangement, and it looks like this. So now this is the central region, and in the central region the central link is joined to the peripheral link by this structure, and this structure is called as the radial spoke. So that means the radial spoke, it joins to the peripheral link to the central link, while the peripheral links, they are again joined by the connectors. These are by connectors. So these connectors are called as the nexin. So these are the connectors, or they connect the peripheral link structure. So now to this peripheral link structure, a structure like this structure is present. Here the 3 dot, if we series, so this 3 dot structure, this is main important structure, that is called as the dynein arm. So this dynein arm, it helps in the, or it releases the energy for the movement of the, for the movement of the organism. So that means for movement, some energy is required, and this energy is released by this dynein structure. So this is the all about the axonym structure. So now come to another structure, that is called as the basal body. So that means, for example, this is the paramecium, this is the cilia structure, and this inside the body or in the cytoplasm structure, the centrioles are present. They are called as the centrioles. So now in the basal body, the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So what is the ciliogenesis? So that means inside of the body, or the inside of not our body, that means inside the, for example, the paramecium or the euglena. So this structure So here the centrioles forms the cilia and the flagella, and the process is called as the ciliogenesis. So during the ciliogenesis, the centrioles are called as the basal body. So this is the again another structure of the cilia and the flagella which is present inside the body or inside the cytoplasm. So now the structure of the basal body is different. Look at the structure, and this is same as present in the centrioles. So first, before going to the basal body structure, first we should know something about the centrioles. So what is the centriole? So for example, in the cell or during the cell division, for example in the metaphase stage, they are the chromosome which is present in the metaphasic plate. So now the spindle fibers are formed here. So this structure is called as the centrosome. So now what is the centriole, what is the centrosome? So now if we see the structure, the centrosome, here it looks like this. So this is the centrosome. So in the centrosome, two centrioles are present, and they are present perpendicularly to each other. So they are called as the centriole. So in one centrosome, two centrioles are present, and they are present perpendicularly to each other here. And besides this centriole, some amorphous structure is present, and they are called as the PCM. PCM means pericentriolar matrix. Just imagine, or just like the cytoplasm. So that means inside the centrosome, two centrioles are present, and they are present perpendicularly to each other, and now the PCM or the pericentriolar matrix are present. So now if we look at the centriole structure, let's suppose this is one centriole. So in the centriole, the structure looks like this. Just imagine. So that means in the axoneme, two structures are present like this here. So that means, for example, this structure, that means one tube and it is joined to the another tube. If we take cross section, it looks like this. So here, three tubes are joined to each other. 1, 2, 3. So just like that, 9 peripheral structure, 9 peripheral structures are present. So in the axoneme, this is the axoneme structure. So now in the basal body, it looks like this. That means this is the axoneme structure, or this is the basal body, which is similar to the centriole. Okay. So in the basal body of the centriole, the same structure like present, but what is the difference in the axoneme? So the peripheral link are called doublet. So they are the doublet. That means they are present two ring structure, but they are the triplet. So that means they are present or the peripheral link same. It is called as the peripheral ring structure, and this is called as the middle part. They are called as the central ring structure, or the central ring or central structure, which is also called as the hub. So in the peripheral ring structure, the triplet microtubules are formed, while in the exonym structure, the doublet microtubule structure are found. And another difference is that inside the central region, there is no microtubule structure. But in the exonym structure, they have two singlet structures are present. So that's why the exonym structure is called as the 9+2 arrangement, and the basal body or the centriole is called as the 9+0 arrangement. And here the difference is the 9 is the 9 are the doublet, and here the 9 are the triplet. This is the main difference between the exonym structure and the basal body structure. So now look at the structure here. So it looks like the bullock cart wheel structure. That means in the, let's suppose this is the bullock cart, this is the one wheel, and inside it another wheel-like structure. So here the centriole is also looks like same as bullock cart wheel structure. So that's why the centriole is also called as the bullock cart wheel. This is the very important, and in the previous exam this question was arised that which structure is also called as the bullock cart wheel structure, and the answer will be the centriole. So this is the all about the structure or the difference between the exonym and the basal body. So the basal body, it is same as the centriole as in the basal body structure. Here the centriole forms the cilia and the flagella, and the process is called as the ciliogenesis. So now this is the centrosome, this is the another centrosome. Here in the centrosome two centrioles are present perpendicularly, here also two centrioles are present perpendicularly. So now actually first in the cell one centrosome are present, but for example in the cell cycle, this is the M phase, this is the G1 phase, this is the S phase, this is the G2 phase. M phase means division phase, that means mitosis or meiosis. G1 means this is the gap 1. This is the S phase, that means here the DNA is synthesized, or the DNA becomes double. So now this is the G2, that means gap 2, that means the gap between mitosis or the meiosis and to the synthesis phase. So during the gap 2 phase, so now the centrosome becomes the double. So the centrosome actually in the cell, the centrosome are present 1. The number of the centrosome are 1, but during the gap 2 phase, the centrosome becomes double, just like the binary fission. So in the gap 2 phase, it becomes the double, and now it comes to one end and another is come to another end, and now the mitosis and the meiosis occurs. So now the spindle fibers are formed. So this is the all about the centrioles or the centrosomes. So now come to what are the function of the cytoskeleton, or what is the function of the microtubule. As I have already discussed, but just as a summary I am telling you that one is the spindle fiber formation. The second one is the formation of axoneme and basal body of cilia and flagella. Third one is it also helps in the cell differentiation. So that means which cell should convert to which cell, and it is determined by the microtubule. Another is it also distributes to the cell pigment. That means, for example, the chloroplast or the melanocyte. And another is it also helps in the cell movement. That means during the gastrulation, one cell goes to another, or one cell again come to here. So this is the cell movement, and this cell movement is occurred by the microtubule, and most importantly it provides the support to the cell or to the organisms. So this is all about the structure of the microtubule. So if we like the video, then please like and subscribe my video. Thank you. The second cytoskeleton is microfilament. So microfilament. So now the microfilament, here the component of the microfilament is actin plus ATP. Actin plus ATP. But in the microtubule, the main component was tubulin plus GTP plus magnesium. But here importantly GTP is present, and here ATP is present, and here tubulin is present, and here actin is present. So this is the main difference between the structure of the microtubule and the microfilament. And you can memorize like here A for actin, A for ATP. If you confuse between the ATP and the GTP. But importantly, ATP is present in the microfilament and the GTP is present in the microtubule. So now, where the microfilaments are found? So firstly, it found in the muscle cell. In the muscle cell, that here, besides the actin, another myosin is present. So myosin along with actin, it forms the myofilament, and this myofilament helps in the muscle contraction or muscle movement. So the muscle contraction is provided by the microfilament, and along with that it also helps in the, or it is present in the microvilli. The microvilli, that means in the small intestine a folded part is present, and that is called the microvilli, and it helps in the active absorption of the food in the small intestine. So it also be provided by the microfilament. So now main functions of the microfilament are, one is it helps in the locomotion. So locomotion means it is present in the pseudopodia. That means in the amoeba case. So here one pseudopodium. Pseudopodia is the plural. The singular is the pseudopodium. So here the pseudopodia, so the microfilament is present, and it helps in the locomotion. The secondly, it also helps in the muscle contraction. muscle contraction. So now third one is it helps in the support to cell. So remember please, all the cytoskeleton, that means microtubule, microfilament, intermediate filament, they have the main function is the support to the cell. That's why the name is the cytoskeleton. The skeleton is for providing the support to the cell. So now it also allow the intracellular movement. Intracellular movement. Intracellular movement means, suppose inside a cell, suppose this protein and it wants to go here. So it can be occurred only by the microfilament. That means microfilament, it helps in the movement of the anything inside the cell. That means it can move the protein, it can move the carbohydrate, it can to move the lipid. So it helps in the allow the intracellular movement. Along with that, it also helps in the streaming movement. Streaming movement. The streaming movement, it is also called as the cyclosis. So what is the cyclosis and the streaming movement? It is simply the movement of cytoplasm. Okay, so the movement of the cytoplasm or the cyclosis or the streaming movement is occurred by the microfilament. And another is that it also helps in the cleavage during cell division in animal cell. So that means during the cell division, first the karyokinesis occur, karyokinesis, and another is the cytokinesis. Karyokinesis is the division of the chromosome. It occurs first. Then the division of the cytoplasm, or called the cytokinesis occurs. That means at the middle, the cleavage occurs, and this cleavage is due to the microfilament. So these are the main function of the microfilament. So now come to the intermediate filament. Intermediate filament. So unlike the microtubule and the microfilament, intermediate filament, they don't have many functions. They have only one function, that is providing mechanical support to the cell. Or cell support, it is the main function of the intermediate filament. For example, the most example of the intermediate filament is the keratin. Keratin, it provides the support. Again, the laminin, it also helps in the support to the cell. So these are the main function of the intermediate filament. That means it only provides the mechanical support to the cell. So these are all about the cytoskeleton, that means microtubule, microfilament and the intermediate filament. If you like the video, please like and subscribe, and tell others to subscribe also. Thank you. Hello everyone, today we will know about the peroxisome. So the peroxisome, it is an organelle. So the peroxisome, it was first discovered in the rodent kidney. And in the rodent, exactly in the kidney and the liver. Kidney and the liver of the rodent, it was discovered in the rodent. So if we see the diameter is 0.7 to 0.7 micrometer. What is the micrometer? 1 micrometer is equal to 10 to the power minus 6 meter. So 0.6 to 0.7 7 micrometer is the diameter of the peroxisome. So the peroxisome is the single membrane bound organelle. What is a single membrane bound organelle? That means, let's suppose inside the cell, let's suppose this is a nucleus, there is some ER present, there is some Golgi body is present, mitochondria is present, peroxisome are also present. So there are the different types of the organelles are present. So what is the single membrane? What is the double membrane? Some organelle is surrounded by a single membrane, but some organelle is surrounded by a double membrane. For example, the mitochondria, chloroplast, nucleus. So these are they are called as the double membrane bound organelle. Why it is called as the double membrane bound organelle? That means the mitochondria, for example, let's suppose this is a mitochondria. We have already studied about this. So this is the outer layer and this is the inner layer, or it is called as the cristae. So that means there are the two layers are present, same like in the chloroplast, same like in the nucleus. So they are double membrane like structure. That means two membranes are present. But in the ER, Golgi, this peroxisome, so they are the single membrane bound organelle. They are the single membrane bound organelle. Why? Because it is surrounded by a single membrane. So the peroxisome, this is the peroxisome structure. This first layer is called as first structure, they are called as the lipid bilayer. That means it is called as the plasma membrane, or you can say that it is the cell membrane. At the core region, this is called as the crystalline core. At the core region, the crystal-like structure is present at the core, and it is called as the crystalline core. So some transport proteins are also present on the membrane. They are called the transport protein, and many enzymes are present. What are the enzymes are present? For example, the cartilage is present, and inside actually the peroxisome, hydrogen peroxide. This is very, very much important, and it is the neat question that the hydrogen peroxide, H2O2, and the H2O2, the hydrogen peroxide is found maximum in the maximum hydrogen peroxide is found in the peroxisome. That means inside the peroxisome, the hydrogen peroxide is found very much, and this hydrogen peroxide, it is that means some toxic, but it has very merit, H2O2, it helps in the cell signaling. It also helps in the lysis of foreign particles, the foreign pathogens. So hydrogen peroxide, this is the merit point. But one demerit point is that hydrogen peroxide is toxic to the cell. So because the hydrogen peroxide is toxic, and the hydrogen peroxide is always generating inside the peroxisome. So to maintain the hydrogen peroxide, a cartilage enzyme is present. So what is the function of the cartilage? The cartilage converts hydrogen peroxide into the water molecule. So hydrogen peroxide is toxic, but if the cartilage converts into the water molecule, which is the neutral thing and which is the best thing to the cell, so that's why the cartilage is present to maintain the hydrogen peroxide. Don't confuse about the absence or the presence of the hydrogen peroxide. Hydrogen peroxide is needed to the cell or to the peroxisome. Why? Because of this function. It also helps in the cell signaling, it helps in the lysis of the pathogens or the lysis of the bacteria and the virus. But at some extent hydrogen peroxide is needed. But because the hydrogen peroxide is continuously producing inside the peroxisome, so to maintain this hydrogen peroxide level, a catalase enzyme is present, and the catalase converts hydrogen peroxide to the water. Another is the urate oxidase. Urate oxidase, which is another enzyme, is present. So it helps in the purine metabolism. So one is the catalase, another is the urate oxidase. They are the main two types of the main enzymes are present. There are the many enzymes are present, but these two are the main. One is the catalase, another is the urate oxidase, and the urate oxidase helps in the purine metabolism. So if you will ask what is the main function of the peroxisome, the main function of the peroxisome is the first one is it helps in the photorespiration. Photorespiration in plants. Okay, this is the plant. In the plant cell, this is the role of the peroxisome. It helps in the photorespiration. But in the animal, it helps in the lipid metabolism. Lipid metabolism. What is the lipid metabolism? Lipid metabolism means after observing the lipid, let's suppose this is the main chain of the lipid. So after observing, during the digestion, the lipid needs to be cut. For example, this one is the another one, this one is another one. So these are the pieces of the lipid is present. A lipid can be formed, and this is need to be absorbed. So this lipid metabolism is called as the process is called as the beta oxidation. And the beta oxidation is occurred in the peroxisome in the animal, not in the plant. In the animal. So in the plant it helps in the photorespiration, and in the animal it helps in the lipid metabolism by the process of the beta oxidation. So this is all about the peroxisome. So if you are watching my video, please like and subscribe. Thank you. Hello everyone. Today we will know about the nucleus. So this is a cell, and inside a cell a main component or structure is present, and this is called as the nucleus. So the nucleus is, we can say that it is a main organelle of the cell. So this nucleus governs all the activity inside the cell. So that's why the nucleus is also called as the director of the cell. Director of the cell. Why it is called the director of the cell? Because it directs or governs the whole total cell. So that's why it is called the director of the cell. So now the nucleus, it was discovered by Robert Brown in 1831. So in 1831, Robert Brown discovered the nucleus. So look at the structure. This is a nucleus. So the nucleus has 4 parts. Mainly it can be divided into 4 parts. The first part is called as the nuclear envelope. Nuclear envelope. The second part, we can say that it is the chromatin material. Chromatin material. The third part is called as the nucleolus. Nucleolus, which is the main component of the nucleus. And the fourth one is called as the nucleoplasm. Okay, these 4 structure or the nucleus can be divided into these 4 parts. One is the nuclear envelope, one is the chromatin material, third one is the nucleolus, and the fourth one is the nucleoplasm. So first come to the nuclear envelope. Nuclear envelope. What is called as the nuclear envelope? It is simply like the plasma membrane or the cell membrane. Look at the structure. This is the cell. So this is the border, or it is confined by a border-like structure, and that is called as the plasma membrane or the cell membrane. So like that, the cell also surrounded by a membrane, and this membrane is called as the nuclear envelope. So it is just like in the cell, the plasma membrane is present. So like that, in the nucleus, the plasma membrane of the nucleus is called as the nuclear envelope. Look at the structure. If I zoom the structure of the nucleus, it looks like this. That means this is the nucleus, and inside it a structure is present, and this is called as the nucleolus. We'll study about that after some time. So first come to the nuclear envelope. Look at the structure. So this nuclear envelope, it is double membrane. Double membrane. But the cell membrane, the plasma membrane, is the single membrane. It is the single membrane structure, but the nuclear envelope is the double membrane structure. So it is the outermost double membrane structure, and look at the structure. This is called as the outer layer. Outer layer, and the inside one is called as the inner layer. Inner layer of nuclear envelope. So the space between outer membrane and the inner membrane is called as the perinuclear space. Perinuclear space, which is the space between inner layer and the outer layer. So, and before that, the nuclear envelope, it is also called as the karyotheca. Karyotheca. So now come to again. So this is the outer layer, and this one is the inner layer, and the space between that is called as the perinuclear space. So this layer is not continuous. So that means, like in the cell membrane, let's suppose this is a cell membrane, this is a cell, it is the cell membrane. It is a continuous, but this membrane is not continuous. That means there is some divided in that, in some area, and this divided part of the pore-like structure is called the nuclear pore. The nuclear pore. And the nuclear pore, if we ask what is the exact size of the nuclear pore, it is nearly 500 angstrom. What is the angstrom? 1 angstrom is equal to 10 to the power minus 10 meter. Okay, so the nuclear pore is the 500 angstrom, that means 500 into 10 to the power minus 10, that means 5 into 10 to the power minus 8 meter. So that means this is 500 angstrom, 500 into 10 to the power minus 10 equal to 5 to the power minus 8 meter. So this is the size of the nuclear pore. So what is the function of the nuclear pore? Look at the structure. Let's suppose this is a nuclear pore-like structure. So that means through this nuclear pore, the RNA and the protein can pass to the cytoplasm, or it can pass from the cytoplasm to the nucleus. So this is the main function of the nuclear pore, that is for the transport of RNA and the protein. The DNA never passes the nucleus, because the nucleus should remain inside the the chromatin or the DNA should remain inside the nucleus. So that's why DNA never passes the nucleus. Only the RNA and the protein will passes to the cytoplasm, or the RNA and the protein from the cytoplasm, it can pass to the nucleus through nuclear pore, which is the size about the 500 angstrom. So this is the outer layer, this is the inner layer. Look at the structure exactly. So at the outer layer a structure is present that is called as the endoplasmic reticulum. So that means, let's suppose this is the nucleus, this is the inner layer, let's suppose this is the outer layer, and from the outer layer the endoplasmic reticulum emerges. So from this outer layer endoplasmic reticulum emerges like this. Look at this structure. So that means the inner layer is present inside, but the outer layer is continuous with the endoplasmic reticulum. That means the outer membrane is connected to the membrane of the endoplasmic reticulum. So it is formed by it forms the endoplasmic reticulum at the outer side. So this is all about the nuclear envelope. So if you are watching my video, please like and subscribe my video. So now come to chromatin material. So chromatin material. So what is the chromatin material? So that means this chromatin material are present inside the nucleus, and this chromatin material is main component of the nucleus, or this chromatin material, because it contains the gene or the DNA. So that means this structure regulates our entire the cell. So that's why they are called as the director of the nucleus or director of the cell. So now simply, if in the exam it will be asked that what is the chromatin material, so simply you can say that the thin thread-like structure that is present inside our nucleus is called as the chromatin material. And always the chromatin material or the DNA, so they all are the negative. So negative means it is the acidic. So because the nuclear material or the chromatin material is acidic condition, so now it is stained by the basic dye. So always you should know that the chromatin material, it is stained by the basic dye, because the chromatin material is itself the acidic, so that it can be only stained by the basic dye. So now the chromatin material, it contains DNA plus RNA plus proteins. So now this protein, it can be the histone protein or it can be the non-histone protein. Okay. So now come to what are the exact structure of the chromatin material. So you already know the word chromatin, chromosome, chromatid and DNA. So what are the chromatin, what is the chromosome, what is the chromatid and what is the DNA? So actually these all structure are the same, but the word the chromatin, chromosome, chromatid in the DNA, it differs because it depends this structure is present in which stage of the cell division. So now you should know something extensive about it. So in the cell cycle, let's say this is the cell cycle, this is called as the M phase. So this is called as the gap 1 or G1 phase. So this is called as the S. S for synthesis phase, and this is the gap 2 or the G2 phase, and it is the G0. Okay. So now these gap 1, S and the gap 2, these are called as the interphase of the cell cycle, while this M phase is called as the division phase. Why it is called as the division phase? Because only in the M phase the chromosomes are divided. So that's why this is called as the division. And why it is called the interphase? Because during the gap 1 synthesis or the S phase or the gap 2 phase, and in these phases the chromosome doesn't divide, it only prepares for the division. So that's why it is called the interphase. So now, what is the structure of the chromatin, chromosome, chromatid? Now we can say that. So when the chromosome is present inside the interphase, that means gap 1 S phase and the gap 2, and this structure now it is called as the chromatin. And this chromatin structure, it is very, very thin structure. Okay. So now, when it enters to the M phase or the division phase, so now this chromatin becomes condensed. What is condense or condensation? What is the condensation? That means simply you can say that the chromatin thickens, and it thickens and it can form a broad structure, and that is called as the chromosome. And now it is called as the chromosome. So that means the structure of the chromatin and the chromosome are the same, but the chromatin is present in the interphase, and during the presence in the interphase, the chromatin is present in the thin, or you can say that it is present in the decondensation phases. That means it is become a very, very thin. So now after going to the division phase, now it condenses and it enlarges or the thickens and to form now the chromosome. So that means the structure, if it is present in the division phase, then it is called as the chromosome, and if it is present in the interphase, then it is called as the chromatin. Okay. So another word is the chromatid. So what is the chromatid? So now look at the structure at the group at the gap 1. The chromatin structure is like that. Okay. In the S phase or the synthesis phase, here the number of the DNA doubles. DNA doubles. So that means here it is called as that's why it is called as the synthesis. That means here the synthesis of the DNA occurs. That means the number of the DNA doubles. So now here number of the DNA doubles, and now it is called as the chromatin structure, and here also it is called as the chromatin structure. But in the gap 1, the chromatin or the DNA or the number of the DNA, let's suppose it is 1, and now the number of DNA is 2. So what is the DNA? What is the chromatin? So first of all, you should know something about the DNA. Look at this structure. So this is the DNA structure. Okay. So actually the DNA structure, this is simply a thread-like structure. So this thread-like structure, it wraps around each other, and now it forms the chromatin structure. So this chromatin, this chromosome, it is simply the DNA. So once this DNA, it wraps around each other to form the chromosome and the chromatin. So in the one chromatin, let's suppose this is the one chromatin. In the one chromatin, one DNA structure is present. That means here this is the chromatin structure. Here one DNA wraps around each other, and now it forms like this structure, and it is called as the chromatin. Look at this structure. This is the DNA. This is simply the thread-like DNA structure. Now it interacts with the protein, and this protein is called as the histone protein. Look at this structure. This is the histone protein. So now the DNA with histone protein is present, and this structure is called as the nucleosome structure. Okay. So now the DNA with histone protein, it forms the nucleosome. Now it again wraps around each other like that. Here it wraps around each other, and now it forms the chromatin structure. Okay, or the chromosome structure. You can say what is the chromatin and the chromosome, but it depends whether it is present in the division phase or in the interphase. If it is present in the interphase, then it is called as the chromatin, and if it is present in the chromosome. If it is present in the division phase, then it is called as the chromosome. So now in the gap one, here the chromatin structure is like that. Okay, but in that case, for example, in the division phase, it is looks like this. So what is the double structure? Why? Because in the synthesis phase, here another chromatin structure or another DNA is synthesized. So now these two chromatins are joined together. Now it forms like this. In the book, you know this structure, and now this is called as the chromatin. So now in the S phase, the chromatin is present. In the gap one, the chromatin is present, but in the gap one, the DNA number is 1. But in the S phase, the chromatin is present, but here the DNA number is 2. Because here one DNA structure, it forms one chromatin, and here another, it forms another chromatin. Okay. So these two, this is the 1 and this is the 2, or here 1, this is the 2 structure, and each one is called as the chromatid. This is the one chromatid, this is another chromatid. That means in one chromatin or the one chromosome, 2 chromatids are present. Here one chromatid, here another chromatid, and each chromatid are called as the sister chromatids. Sister chromatid. Okay, so now this is the one chromatid, this is the another chromatid, and they are called as the sister chromatid. So if we look like this structure, at the center one, it is called as the centromere. Centromere. Okay, so now in the centromere of the each chromatid, there is a one structure is present, and this is called as the kinetochore. Okay, here also one kinetochore is present. So what is the function of the kinetochore that is present in the centromere? It helps in the binding of the 2 chromatids, because this kinetochore that is present in the centromere, that helps in the binding of the chromosomes, and now after this it forms like this structure. So that means the 2 kinetochore that is present, and they helps in the binding of the chromosome. So now this is called the chromosome, and this is called the centromere. So now it contains 2 kinetochore. That is very much important. Kinetochore. So that means 2 kinetochore are present in the single chromosome structure, because in each chromatid 1 kinetochore is present. Okay. So this is the main structure of the chromatid, chromosome, chromatin, and the DNA structure. So now if we look at the chromatin structure, the chromosome structure, let's suppose this is the chromosome structure. This is the one sister chromatid, or it is called the chromatid, one chromatid. This is another chromatid, or simply they are called as the sister chromatid as present. And here one kinetochore is present. Here also one kinetochore is present. That means here the 2 kinetochore as present, and this kinetochore that helps in the binding of the 2 chromatins. Okay. So now look at the structure of the chromosome, and here one constriction is present, or this is called the primary constriction, and this primary constriction is called as the centromere. Okay, so this is another constriction is also present here, and this is called as the secondary constriction, or it is can called via the satellite Okay, the first constriction, because it is very much it is inward towards the center, it is called the primary constriction and it is called as the centromere, and the secondary constriction, it is here present and now it is called as the satellite. So now look at the structure of the chromatin, that means how many types of the chromatins are present, or if we divide the chromatin, it is two types. One is called as the heterochromatin, another is called as the euchromatin. So one is called as the heterochromatin, another is called as the euchromatin. Look at the structure of the chromosome. So here, this dark color, this is the dark strand, they are the dark strand, they are the dark strand, and they are called as the heterochromatin. Okay, so now we can say that the heterochromatins are darkly stained. While the euchromatin, this is euchromatin, they are the euchromatin. So they are the euchromatin, or it is, you can say that they are lightly stained structure that is present inside the chromosome. So what is the function of this heterochromatin, what is the euchromatin? So first come to the heterochromatin. So the heterochromatin, here the genes the present inside the heterochromatin, here and here, the genes present in the heterochromatins are silenced. So what is the silenced DNA? So that means here the gene that is present, or the DNA sequence that is present in the heterochromatin is silenced. So that means it is not transcriptionally active. That means they are, or the genes are not transcripted. That means the transcription never takes place in that gene that is present in the heterochromatin. So that means here that is called as the silence. That means there is no protein is synthesized from this gene present in the heterochromatin. While in the euchromatin, they are very active. That means all the protein are synthesized. After transcription, the mRNA is present, and from the mRNA, the proteins are present. The proteins are formed. So that means all the proteins, or the maximum of the proteins that are formed, or the maximum of the genes that are transcribed, that are comes from the genes that are present in the euchromatin. So that means it is called as the actively genes. That means here always the transcription takes place. So that's why they are called as the silent gene or inert genes, or the inert activity occurs in the heterochromatin, while the euchromatin, there the active genes are present. So now, because there is a silent gene, for example, this is the nucleus. So the heterochromatin, because it is the silence of the inert structure, so they are always present in the periphery region of the nucleus. So they are present in periphery region of nucleus. While they are present, euchromatin, they are present at the center phase. That means at the central part, the euchromatin are present, while at the periphery region, the heterochromatin are present. So now, the heterochromatin, they are present only in the eukaryotes, while the euchromatin, that is present in both prokaryote and eukaryote. Okay. So now the heterochromatin, it can be divided into two parts. One is called as the constructive, another is called as the facultative. Facultative heterochromatin or the constructive heterochromatin. So what is the constructive heterochromatin? That means here the genes are always or the permanently inactive. That means the genes that are present in the heterochromatin, there is permanently inactive. That means there is never transcription occurs in this constriction of heterochromatin. But in the facultative, that means they are always or maximum case, the maximum time they are present in the inactive condition, but sometime it transcribes. Okay, what is the example of the facultative heterochromatin? One example is the bar body. The bar body is present, and that is the facultative heterochromatin. That means it can transcribe if needed. So this is the main difference between the euchromatin and the heterochromatin structure. Now come to most important portion of the nucleus, and that is called as the nucleolus. So what is the nucleolus? First of all, the nucleolus, it is a singular term. If more than one nucleus is present, then it is called as nucleoli. And the nucleoli, it is the plural term. That means if more than one nucleus is present, then it is called as the nucleoli. Otherwise, it is called as the nucleolus if it is present in the one number. So this is the structure of the cell. Let's suppose this is all over. It is called as the nucleus. Okay. So now inside the nucleus, at the middle portion, there is a portion like this, or there is a structure like this, and this structure is called as the nucleus. Okay. So nucleolus. So nucleolus, it was discovered by Fontana. In 1971. So, and he discovered in eel skin. That means eel is a fish. So, in the eel skin, the Fontana discovered the nucleolus. So now come to the structure of the nucleolus. So if you ask what is the structure or the exact structure of the nucleolus, then it is a round color, round, dense and darkly stained structure that is present inside the nucleus is called as the nucleolus. So now the nucleolus, look at the structure. The nucleolus, most importantly, it doesn't have any membrane. So that means most importantly, nucleolus is a non-membrane structure. That means it doesn't have any membrane structure. So in some organisms, the number of the nucleus is 1. That means we can say that there is a nucleus. For example, in our case, for example, in the human case, so the number of the nucleolus is 1. So you can say that it is the 1. But in many organisms, there are many nucleolus. So we can say that they have nucleoli because it is a plural term. So that means if the number of the nucleolus is more than 1, then it is called as the nucleoli. So now this nucleoli structure or the nucleolus, because it doesn't have any membrane, so now the content of the nucleus, For example, these are the content of the nucleus and they are called as the nucleoplasm. So what is the nucleoplasm? Nucleoplasm. So what is the nucleoplasm? It is just like the cytoplasm. For example, inside the cell. So they are called as the cytoplasm. So like that, inside the nucleus, the cytoplasm is called as the nucleoplasm. So because the nucleus is a non-membrane structure, that means it doesn't have any membrane, so that the content of the nucleoplasm is continuous with the nucleolus. That means the content can easily go to inside to the nucleus of the nucleoplasm, or the content present in the nucleoplasm, it can transport to the nucleolus freely because it doesn't have any membrane structure. So now, how the nucleolus is formed? So most importantly, how the nucleolus or this structure, how it is formed. So nucleolus is formed by most important, that is called the NOR. NOR means nucleolar organizing region. So that means, so inside the nucleus many chromosomes are present. So around particular chromosomes, so the nucleolus is formed. For example, let's suppose this is a chromosome. So in the chromosome, around the chromosome a structure is formed, and that is called as the NOR, the nuclear organizing region, and it is called as the nucleolus. And it is, one is called as the, this is called as the centromere, or it is also called as the primary constriction. Okay. So now, around this part, it is called as the secondary constriction. So that means the nuclear organizing center is simply, you can say that it is the secondary constriction, and the nucleolus is formed around particular chromosomes, not one. Normally 1 to 6, around 1 to 6 chromosomes. For example, this is one chromosome, this is another chromosome, this is again around, this is another chromosome, this is another chromosome, this is another chromosome, this is another chromosome. So around this, always at the secondary constriction. So by combining this secondary constriction of the NOR region, a nucleolus is formed. So if you ask in the human, around 5 chromosomes, the nucleolus are formed. And if you ask the exact what are the 5 chromosomes around which the nucleolus or the nuclear organizing region is formed, and the answer will be 13, 15, 20, 21 and the 24th chromosome. That means around these 5 chromosomes, the nuclear organizing region. So 24 means out of 46. So 13, 15, 20, 21, 24, around which this nuclear organizing region or the nucleolus is formed. Okay. So now come to the what is the exact structure of the what are the parts of the nucleolus. So there are the mainly 4 parts are present. One is called as the granular zone. The second one is called as the fibrillar zone. The third one is called as the amorphous, or simply you can say that the matrix, or you can say that pars morpha. And the fourth one is called as the nucleolar chromatin. Most importantly, it is the nucleolar chromatin, not nuclear chromatin. Nuclear chromatin means the chromosome that is present in the nucleus. So the nucleolar means the nucleoprotein or the nucleic protein or the rRNA, more exactly the rRNA that is present, and that is called as the nucleolar chromatin, not like the DNA that is present in the chromosomes. So that is present in the nucleus, but in the nucleolar or the nucleolus, there the rRNA is present. So first come to the granular zone. Look at this structure. This is the structure of the nucleolus. At the outer chromatin material, they are called as the perinucleolar material. That means the chromatin material that is present outside the periphery region, it is called as the perinucleolar material. And all the materials, nuclear material that is present inside it, it is called as the intranucleolar chromatin. That means they are present inside or the center of the nucleolus. So first structure is called as the granular zone. Look at this structure. This is the granular zone. So always the granular zone, it is present in the periphery. It is present in the periphery region. So what is the granular part, or what structure that contributes to the granular structure or the granular zone? Mostly it is ribonucleic protein. Ribonucleic protein. So what is the ribonucleic protein? Simply you can say that the rRNA and the associated protein is called as the ribonucleic protein. So at the outer region or the periphery region, the granular portion is present, and it contributes, or it contains the ribonucleic protein that contains rRNA and its associated protein, and it is called as the ribonucleic protein. The second one is called as the fibrillar zone. Look at this structure. This is the fibrillar zone, and as the name suggests, the fibril or the fibrilla, that means the fibril-like structure that is present inside the nucleolus, and they are present inside or the center part, which is called as the granular portion. So now it contains rRNA plus proteins, and that is the non-histone protein. Non-histone protein. So in the NEET exam, sometimes it will ask that in which part of the nucleolus the proteins are present, or the ribonucleic protein. If the ribonucleic protein is not mentioned, it is asked that in which part of the nucleolus the proteins are present, the answer will be the granular zone. And if it will ask that in which part the rRNA is present, the answer will be fibrillar zone. Okay. The third one is the amorphous or the matrix or the pars morpha. So look at the structure like the cytoplasm, like the structure that is called the middle part, or simply it is called as the matrix, or the inside or the center, the exact center, it is called as the amorphous or the matrix of the Mars morpha. And the fourth one is the nucleolar chromatin. So the nucleolar chromatin means here the rRNA or tRNA. So these rRNA, tRNA are mainly present in the nucleolar chromatin along with some DNA structure. Okay. So now what is the function of nucleolus? So if nucleolus is present, so what is the main function of the nucleolus? The main function of the nucleolus means it provides machinery for the protein synthesis. Machinery for protein synthesis. How? So that means if the protein means the transcription is required, transcription, and then translation is also required. So during the transcription and the translation, a rRNA or the ribosome is required, and these rRNA and the ribosome are actually formed in the nucleolus region. So that's why it helps in providing the machinery for the protein synthesis. Which machinery? That is the rRNA and the ribosome. The main second answer is that it synthesizes simply the rRNA and ribosome. So in the NEET exam, it is always asked that what is the site of rRNA in the ribosome? The answer will be the nucleolus. If in the option it will be as that nucleolus, both. The answer will be nucleolus, but because exactly inside the nucleolus, the rRNA and the ribosomes are formed. So it is the site of the rRNA in the ribosome synthesis. So third one is it also stores the ribosomal protein. Ribosomal proteins. Okay, so these are the main function of the nucleolus. So if you like the video, please like and subscribe. And next come to the nucleoplasm. So nucleoplasm. So what is the nucleoplasm? Simply the cytoplasm-like structure that is present inside the nucleus is called as the nucleoplasm. Look at this structure. This is the nucleus, and inside it the cytoplasm-like structure or the watery-like structure is present, and this water-like structure is present, and that is called as the nucleoplasm. So the nucleoplasm, it is also called as the nuclear sap. Nuclear sap, because it is the sap-like structure that is present inside the nucleus. That's why it is also called as the nuclear sap, or it is also called as the karyoplasm. So karyo means nucleus and the plasm means like the cytoplasm-like structure, and because the cytoplasm-like structure that is present inside the nucleus, that's why it is also called as the karyoplasm. So the nucleoplasm, it is simply the matrix structure. That is, matrix means the inside structure. So that's why it is simply also called as the nuclear matrix. That means the structure that is present inside the nucleus. So the nucleoplasm contains chromatin and nucleolus. So this is the nucleolus and the chromatin material that is present inside it, that is called as the chromatin, and that is the main difference between the cytoplasm and the nucleoplasm. So that means what is the cytoplasm and what is the nucleoplasm, or what are the difference between them? So the cytoplasm and the nucleoplasm, they contains mostly the watery substances, and beside the watery substances, the nucleoplasm contains the chromatin, and this chromatin structure that is present inside the inside the nucleoplasm, and that's why it also helps in the, because of the presence of the chromatin, it also helps in the formation of DNA and the RNA, which is absent in the cytoplasm. So that is the main difference between the cytoplasm and the nucleoplasm, that the chromatin material is present inside the nucleoplasm, and that helps in the formation of the DNA and the RNA, which is absent in the cytoplasm. This is the main difference. Another difference is the cytoplasm is less sticky, while the nucleoplasm is highly sticky or highly gelatinous. What is the gelatinous or the highly sticky? That means it is present in a more concentrated manner in comparison to the cytoplasm. So they are the two difference between the cytoplasm and the nucleoplasm. One is the presence of the chromatin in the nucleoplasm. Another is it is the highly sticky or the highly concentrated structure in comparison to the cytoplasm. So they are the main difference between the cytoplasm and the nucleoplasm. So besides the nucleoplasm or the DNA, also the RNA are present inside the nucleoplasm, and it contains many hydrolytic enzymes. That means the nucleoplasm it also contains many hydrolytic enzymes, hydrolytic enzymes. So hydrolytic enzymes means the main difference or the main function of the hydrolytic enzymes means they cleave the biomolecules. For example, the peptidase. The peptidase means it cleaves the protein or the peptide bond. That's why it is called the peptidase enzyme. The peptidase enzyme, the function is to cleave the protein that is present inside the nucleus. Another is the ribonuclease. Ribonuclease means to cleave the nuclear bond. The nucleus, that means DNA RNA bond, that means to cleave the phosphodiester bond, that means to cleave the DNA and the RNA. The hydrolytic enzymes are also present, and the main important enzymes are also present, that is called as the DNA polymerase. So DNA polymerase, its main function is to, that is the DNA replication. So the DNA replication enzyme, DNA polymerase, is also present inside the nucleoplasm that helps in the replication of the DNA. So this is the main important function of the nucleus, and another function of the other function are also present. For example, many metabolic activities are also takes place in the nucleoplasm, and again most importantly it also helps in the maintenance of nuclear shape. That means it also helps in the maintenance of the nuclear shape. That means the nuclear shape is also maintained by the nucleoplasm. So this is the main function and the main structure of the nucleoplasm. So if you like the video, please like and subscribe my video. Thank you. Hello everyone. Today we will know about the chromosome. So in this video we will know about the what are the different types of the chromosome that are present and what is the role of the chromosome in the organisms. So before going to the chromosome, we should know something about the chromatin. So you have already heard the term chromosome. You have already heard the term chromatin and the DNA. So what are the difference between this chromosome, chromatin in the DNA? So now get into this. So in the cell cycle, let's suppose this is the cell cycle. So in the cell cycle, there are the different types of the phases are present. So the first stage is called as the gap 1 phase, or it is also called as the G1 phase. So that means, and before that another stage is called as the G0 or the quiescent stage. So that means if any cell, if it wants to go for the division, then it transfers from the G0 to the G1. If it doesn't want to divide, then it remains in the G0. So that's why the G0 is called as the quiescent stage. So now if the cell wants to divide, then it left the G0, then it transfers to the G1 and the gap 1. Then after the gap 1 there is a phase is called as the S phase. S for synthesis. So what is the synthesis, or why the name is called the S phase or the synthesis phase? Because here the what does the it synthesize? It synthesizes the DNA. So that's why it is called the S phase. So after the S phase, another phase is called as the gap 2 or G2 phase, and after that, that is the main phase that is called as the main page or the M page, or it is called as the mitosis or the meiosis page, because in this page actually the cell division occurs. So the gap 1 S page, gap 2 is called as the interphase, and this is called as the M page is called as the division phase. So what is the difference between the interphase and the division phase? Because in the interphase or during the interphase, the cell already prepares for the division, but in the interphase there is no division in the cell. The cell divides only in this M page, but in the interphase it prepares for the division, so that's why it is called the interphase. Okay, so now come to the term the chromosome, chromatin and the DNA. So what is the DNA? So DNA, simply imagine a thread of simply you can imagine like the thread. The thread is like this. So if this thread, it coiled together one and more, and now it forms a structure like this, and this is called as the chromatin. Okay, so in the chromatin at the middle it is called as the centromere. So that means our centromere, it is also called as the primary constriction, that divides these two arms. This is the one arm, this is the another arm, and this is called as the chromatin. So that means in the gap 1, the structure of chromosome, it looks like this, and this is called as the chromatin. Okay, so that means what is the difference between the DNA and the chromatin? That means the single DNA or the one DNA coiled together each other, and it forms the chromatin like structure and looks like this. That means one chromatin, we can say that one chromatin contains one DNA. So that means at this page, that is, there are 23 DNA are present. So that means the 23 chromatins are present. So in our cell, for example, in the animal cell, the 23 DNA are present, and this is called as the chromatin in the gap 1 page. Okay. So now in the S page, the DNA is synthesized. So that means another DNA, that means in that chromosome, one DNA is present, and it coiled together to form this like structure that is called as the chromatin. So in the S page, one chromosome or the one DNA replicates, and now two chromosomes